I think that when we are ready, we can start.
Okay.
Yeah.
Khalil, would you like to start us?
Professor Khalil can start. Yeah.
Thank you very much. On behalf of myself, I would like to welcome you all. It's really nice to be back after 2 years of scientific limbo. And we have been very afraid to come to Nairobi because we were afraid that the COVID-19 will be lingering in the air. Unfortunately, we were not able to make the journey. So we felt a bit relieved. My name is Tahir Khalil, and I'm a professor of hematology and tropical medicine at the Institute of Endemic Disease. And in fact, I was really happy to hear in the last 2 days about control and elimination, because this region has a wealth of data about vaccines from Ishmaniasis. About 3 decades ago, we started a WHO TDR-sponsored study on vaccine for visceral Ishmaniasis. It was a phase 3 study that came to conclusion. And following that, we predicted the first in-Situ molecule that can be used as vaccine for visceral Ishmaniasis. And currently, Professor Mosa, in collaboration with Professor Paul Kay, they are carrying cutting-edge research on vaccines for Ishmaniasis. So it's really a good thing that we are going back to looking into vaccines. And in this session, we'll be talking, people will talk about the immunology of chronic VL, HIV co-infection, immunology of L infantum and L major. And the first speaker will be Dr. Nikki DeBrech. I hope I pronounced his name correctly. Nick works as a PhD researcher at the Clinical Immunology Unit of Antwerp Institute of Tropical Medicine and the Adam Data Lab at the University of Antwerp. Here he focuses on the T-cell driven immunity observed Ishmaniasis and the entire process of antigen presentation and recognition. Besides this, he is working on analyzing the immunological sub-study of the predicting visceral Ishmaniasis in HIV patients clinical study. Here, they are trying to characterize host immunological markers VL HIV infection and profile the evolution over time to find determinants of VL relapse. Nick carries out his work at the interface of two complementary skills, bioinformatics and biomedical sciences, using computational approaches to tackle complex pathogens and infectious disease. And the floor is yours.
Okay, hello everyone. I'm Nick de Vrij. Today I'll be speaking about the immunological determinants of relapsing visceral leishmaniasis in co-infected patients. So to give you a little background here, VL HIV co-infection is very prevalent, especially in the region where we conduct our pre-leash study, which is Ethiopia. I think a large majority of VL patients also have HIV co-infection. I would say up to half of these patients. And these patients follow a really chronic disease course. So they relapse very frequently and they cure intermittently, but they will keep relapsing despite the fact that they are fully virologically suppressed by antiretroviral therapy. So what we try to do in the pre-leash study is two things. First, predict the VL symptom development or VL infection development in HIV patients, but also to try to characterize what are some immunological determinants of relapse cases. So prior studies have already tried to do this, and they tried to find some CD4 to find some predictor markers, for example, CD4 count, and they found that CD4 count at baseline can already predict VL relapses. And here we did not find the same thing, which I'll come to later. So the PRELISH study is a consortium between the University of Gondar, the Institute of Tropical Medicine, and Doctors Without Borders. It was situated in Abdurrafi in Ethiopia. And the aim here was to predict visceral leishmaniasis development in HIV-infected patients by studying the asymptomatic period prior to the onset of active disease in HIV-infected individuals that were monitored over time, and were also monitored post-treatment to see if they relapse or not, or if they cure. And this was a very unique cohort of 570 patients, HIV patients, of which around 34 developed symptomatic VO HIV co-infection. So in this sub-study, this was really focused on the immunology of VO HIV patients, contrary to the bigger PRELISH clinical study, where they focused on a lot more molecular markers as well. So this is all still very preliminary. The study is not entirely done yet, and we are still working on the data. So the aim of this sub-study was really to describe the evolution of human host immune markers over time to identify patterns of these markers that were associated with VL relapse. An idea about how the study looked like. So we took around 24 symptomatic VL patients, 21, that we sampled, not the entire 34, because these were recruited later. And these were monitored from baseline until study end. So this is what you see here. We have 14 relapse cases on the top and 7 relapse case, 7 curing patients at the bottom, and these were all sampled at several intervals. For example, at day 0, which is the active disease, the active VL disease development, during the 4 weeks of treatment until end of treatment, where they were also regularly sampled. And then afterwards, they were monitored for a long period, up to 28 months. For one of these patients, where they either relapsed or did not relapse, and samples were regularly taken. So what did we do with these symptomatic VL HIV patients? We used flow cytometry on peripheral blood monocytes derived from these patients to study a panel of cytokine markers, T-cell activation markers, T-cell senescence, and exhaustion, immune exhaustion markers. But added to that, we also, to look into the immunity and the immune profile of these patients, we also looked in a more unbiased way, without being limited or restricted to these pre-selected markers. So we applied single-cell RNA sequencing and simultaneously T-cell receptor profiling on a subset of these patients. So we didn't only do this for symptomatic VL HIV patients. We also did this on the regular HIV-infected patients that were truly negative for any leishmania infection. And we also applied this on asymptomatic leishmania HIV co-infected patients. And for the single cell sequencing, also on 2 healthy endemic controls. So to give you a little bit of an insight about how these patients look like, what these patients look like. All of these patients, all of the symptomatic VL-HIV patients, contrary to the HIV patients, these patients were all male. And these were all relatively young, young patients. The relapse VL-HIV patients were slightly younger than the cured patients, which we didn't expect at first. But in terms of their occupation, there's really not a big difference. Most of these were daily laborers or farmers working in the fields. So to give you a little bit more insight on their clinical features, at recruitment baseline, there was no difference in their CD4 T cell count. And at active disease, there was also no difference in their CD4 T cell count. But what we see at the post-month 6 time point is that the CD4 T cell reconstitution is happening in curing patients, but not in relapse cases. So we see this on the right here, where the cured patients have a much higher CD4 T cell count than the relapse cases, making it a possible important predictor for relapse cases, except for the fact that this is at the post-month 6, so 6 months after treatment time point, and not at day 0, which means it can't be truly used as a predictor, contrary what other studies have tried to do or have found, is that the CD4 count at baseline is already a predictor, which was not the case here. So we knew these CD4 T cells were, of course, very dysregulated, as happening in HIV alone as well. So we tried to look farther into these CD4 T cells and see what their profile, immune profile looked like. So we looked at the proportion of CD4 T cells that produces interferon gamma. And we found that at day 0, so at the active disease development stage, the proportion of interferon gamma producing T cells, CD4 T cells, was actually higher in the curing patients than in those that relapse. And this was not only the case for the active disease development time point, but this was the case for the entire disease course. So starting from the asymptomatic period up until the post-treatment monitoring phase, the proportion of CD4 T cells that produce interferon gamma was actually a lot higher, meaning that, especially because it already happens in the pre-treatment phase, it could potentially be used as a predictor for VL relapse. So we did not focus only on interferon gamma production. We also looked at the immune exhaustion markers. So this is just one example of an exhaustion marker, which is the proportion of CD4 T cells that produce PD-1, a known exhaustion marker. And this is shown at the top. And even here, the proportion of PD-1 expression in CD4 T cells is actually lower in cured VL-HIV patients as compared to those that relapse. So the patients that relapse really show a more pronounced immune exhaustion. This is also not the only marker where we see this. We see this in some other markers. And we also see this on CD8 T cells, but this has not been shown in my presentation. I will mostly focus on the CD4 T cells. And we also tried to profile some senescence markers. And here there was actually no difference over time or no difference at any time point. So this really shows that the CD4 T cells become more immune exhausted in the relapse cases, and that it's definitely not senescence that's happening. So Next, we try to look at this in a more unbiased manner by applying single cell sequencing. So at the top, you see 2 stages for every disease presentation. So the day 0 in the top left for the cured patients and the end of treatment right next to it, and then moving up to the relapses, day 0 and the end of treatment. So at the top, you see the single cell RNA profiles, where you see the distribution of the different cell types. And at the bottom, you see the T cell expansion or the clonality of T cells. And what this shows at the bottom is that in the curing patients, after treatment, you see a lot more expansion. You see some hyperexpansion of T cells going on. And if you look closely, these are mostly CD4 effector memory cells and CD8 effector memory cells that become expanded upon treatment. But if you look at the relapse cases at the right half or the right quadrant, you see that upon day 0 and end of treatment, there's almost no difference in T cell expansion, and there's almost no expansion going on, which probably means that the relapse patients the T cells of these patients do not get stimulated or are anergic. Okay, the slide can move. Sorry, I don't have control here. Sorry, can someone move the slide? Okay. Maybe I should share my own screen if that works. But yeah, sorry. Okay, so in here, still looking at the single cell RNA sequencing profiles, we try to find some differentially expressed genes in the CD4 T cells of cured VLHIV patients and those that relapse. So these are heat maps, the first one showing the day 0 time point cured versus relapse, and the bottom showing the end of treatment time point. So what this shows, if you look at the difference here, is that the treatment will upregulate the interferon gamma response. And also a new pathway seems to be enriched, which is T cell activation, which closely mimics the T cell expansion we saw in the earlier slide. And also some other pathways related to T cell activation, so regulation of T cell activation, but also antigen processing and presentation, which becomes more upregulated at the end of treatment time point. So after treatment, the T cells and the antigen presenting cells become more activated. And the next slide can be shown. Okay, so some of the things I want you to remember from this presentation, it's that the baseline CD4 count, contrary what has been shown in the literature up till now, is not a good predictor of VO relapse, at least not by its own. Maybe if combined with some other predictors, it could be a good one, but on its own, in our sub-study at least, it does not show any difference between those patients that cure or relapse. What we did not do was profile the CD4 increase after treatment. We only have it for the post-month 6 phase and not at the end of treatment. So it might be that the inability to increase CD4 T cell count after VL treatment, that this might still be a good predictor for relapse. But we have not tested this. So what we did test was and observed was that the CD4 T cells of relapsing VLHIV patients have a more immune exhausted phenotype, and that in the curing patients, there's actually a higher proportion of CD4 T cells that produces interferon gamma, which might be an important predictor. And also these CD4 T cells, they become a lot more activated in curing patients than in those that relapse. So it might really be that the disease-specific or leishmania-specific T cells become activated in cured patients and are anergic or inactivated in the relapse patients, and that this activation might induce them to produce interferon gamma to combat leishmania. So I've shown you only CD4 data right now, but we also have data on CD8 cells, CD8 T cells, NK cells, and antigen-presenting cells, but these have not yet been fully analyzed, and we are still busy with that. I'd like to reinforce that this is preliminary data I'm showing and not the full data yet. And the next steps here will be to train predictive models for the features I've shown you. And with that, I'd like to thank all of the people that helped during this pre-leash study, from both the Institute of Tropical Medicine, but also from the University of Gondar, Doctors Without Borders, and of course the ADFREM Data Lab. Thank you a lot for allowing me to speak.
Thank you very much, Nikki, and we hope to see the results on CD8 and NK cells soon. Our next speaker will be telling us about the immunology of L infantum and L major patients, infections. The next speaker is Dr. Sraya Boussafara at the Institute Pasteur, Tunisia. Dr. Sraya is a PhD holder and associate professor in the laboratory of transmission control and immunology of infection at Pasteur Institute of Tunis, Tunisia. Her main interest is immunology of leishmaniasis, with special emphasis on the immune correlates of protection and characterization and validation of new vaccine candidates for both parasite and xenovirus. The floor is yours, Dr. Frey.
Thank you, Professor. First of all, I would like to thank organizers from LEAP and especially Professor Wazuna for the invitation. And I would like to say that it's a pleasure for me to participate to this meeting. Here I will present some results of our research activity dealing with the study of human response during cutaneous leishmaniasis due to L major and L infantum. So like most region of North Africa, Tunisia is the endemic region for VL and CL. But nevertheless, the cutaneous form is the most prevalent form and largely distributed in the country, which caused a serious public health in Tunisia. So there is 3 forms of cutaneous leishmaniasis. We have the zoonotic cutaneous leishmaniasis in the center. Sorry, I don't have the hand for that. And it's located in the center of Tunisia, and actually it's spreading to the north. Different form of cutaneous leishmaniasis, zoonotic cutaneous leishmaniasis is recorded. Next, please. The second form is the sporadic cutaneous leishmaniasis, which was detected in the north of, which is detected in north of the country, and it's usually detected as a single ulcer in the face of the subject. And actually it's spreading to the center of the country. The third form is chronic cutaneous leishmaniasis, CCL. It's due to Leishmania kiliki. It's discovered for the first time in macrofocus in the southeast of Tunisia, in the region of Tatawin. But actually, we know that it's spreading to the north and the center of the country. It's due to the infection with kiliki. So next slide, please. Sorry. So 3 leishmaniasis species are involved in this 3 forms of cutaneous leishmaniasis. We have the L major, MON25, which is the spread in the north, in the center of Tunisia. We have the L infantum, MON1, MON24, and MON80. It's the modem of L infantum, which discovered in the north and in center of Tunisia. And finally, we have the L kiliki MON8, which is responsible of the chronic cutaneous leishmaniasis. It's true that we have a difference between the number of cases of cutaneous leishmaniasis because the zoonotic cutaneous leishmaniasis is more spread in Tunisia, between 2,000 and 3,000 cases per year. For the sporadic cutaneous leishmaniasis, we have between 20 and 30 cases. And for chronic cutaneous leishmaniasis, we have just 10 cases per year. Next, please. So as we can see, this different form of cutaneous leishmaniasis are due to different factors, among them the virulence of the parasite, the species of leishmania enrolled, and the host human response. For this reason, we conducted a study of human response, local and systemic human response within this CL. So next, please. Next slide, please. This was done using CL biopsies collected from sporadic cutaneous leishmaniasis and zoonotic cutaneous leishmaniasis, and we did an histologic immunologic study for this biopsies. So next, please. Histologic study showed that there is differences between the 2 forms of CL, and we know that the infiltrate is more important within zoonotic cutaneous leishmaniasis. In contrast, the infiltrate is more organized with macrophage and lymphocyte within the infiltrate, within the dermis infiltrate. So next, please. Next slide, please. So the density of the infiltrate was confirmed by, and the presence of macrophage and lymphocytes were confirmed by immunohistochemical analysis, and we showed that the number of the percentage of CD4 and CD8 are more important within sporadic cutaneous leishmaniasis. And when next slide, please. And when we analyze the difference between the percentage of these cells within early or late zoonotic cutaneous leishmaniasis, we found that these cells, CD8 plus T cells, were more important within early zoonotic cutaneous leishmaniasis, having a duration of less than 1 month. So next, please. We tried to analyze the human response more and to identify the phenotype of these T cells. And for that, we analyzed the expression of interferon gamma, IL-10, IL-8, and some chemokine, IL-8 and MCQ1. So as we can see, most of lesion express interferon gamma and IL-10. And when we calculate the ratio interferon gamma IL-10, we found that it's more interferon gamma. There is more interferon gamma within lesion compared to IL-10, indicating probably the Th1 response. So this the expression of IL-10 correlate negatively with the lesion age. You can see in the right of the slide. So next, we analyze the expression of IL-8 and the MCP1, and we see that there is all of lesion express this chemokine, and there is a correlation between them. So interestingly, we see that IL-8 is more expressed within sporadic cutaneous leishmaniasis and MCP1 with zoonotic cutaneous leishmaniasis. Next slide, please. So after that, we try to evaluate the involvement of cytotoxic cells. And for that, I have to say that when we did the immunohistochemical analysis, we didn't find natural killer cells. We found just CD4 and CD8. And when you analyze the expression of granzyme B and granulocyte, we found that most of this lesion express these molecules. And this expression of a granzyme B correlate negatively with the age of lesion. And this is clear when we categorize lesion in early and late lesion. So we found that granzyme B and granulocyte are expressed within early zoonotic cutaneous leishmaniasis, for which we have 1 month duration, less than 1 month duration. Next, this can explain the participation of granzyme B, this pathway, perforin, granzyme B, and granulocyte in the killing of parasite. So perforin will give the possibility for granzyme B and granulocyte to enter the cell, and then granulocyte will permit the entry of granzyme B to the cell, to the parasite, and killing it before killing the cell. So it's the microptosis of granzyme B and granulocyte, and microptosis of the parasite. So next please. We did double staining for granzyme B and interferon gamma, and as you can see in the slide, we showed that granzyme B is mostly expressed by CD8, but interferon gamma is produced by both CD4 and CD8. So here we showed that CD8, that granzyme B and cytotoxic cells are CD8 plus T cells. Next, please. So as we can see, we showed that we found high level of granzyme B and granulocyte mRNA within ZCL and SCL lesion, indicating the active process of parasite killing. But we have to say that the role of CTL during LC still be controversial. And we ask the question, what is the role of these cytotoxic cells in resistance to reinfection? So for that, we conduct a prospective study. Please, the next slide, please. Next slide, please. So resistance to Leishmania major is attributed to development of Th1 response with proliferation, production of interferon gamma, and Leishmanian skin test, positive Leishmanian skin test. In contrast, we have demonstrated the involvement of cytotoxic T cells, and we show the presence of production of granzyme B by PBMC stimulated with metacyclic promastigote of Leishmania major within patient, ZCL patient with healed ZCL individual and some healthy individual living in endemic area for transmission of L major. Next, please. Next slide, please. Sorry.
Just there's a slight delay, so it's expected to come up in a moment.
Yes, actually, I have two slides, two other slides. Sorry, but can I get the next slide? No?
I think there is a problem with the internet.
I think there is some problem.
I think there's a problem with the internet. I think give them like 1 minute and they should be able to resume.
Okay.
Yeah.
It's okay. Okay. Can I go?
Yes, yes, yes.
Previous slide, please. Okay, so we conducted a prospective study of cohort of 453 exposed subjects living in endemic area, and during the first visit in the field, we did a clinical exam. We analyzed the leishmaniasis skin test response, and we get blood sampling for analysis of granzyme B production and some other parameter. Next slide, please. We showed that there is correlation between the granzyme B production indicating a cytotoxic immune response and the leishmaniasis skin test, but some discrepancy were observed within this between these two parameters, and we show that the higher level of granzyme B are found within LST positive individual and those having a scar, ZCL scars. So the second visit in the field allowed us to diagnose the new cases of ZCL, and based on the immune response collected and detected at the baseline, we analyzed the effect of immune response on the protection against development of leishmaniasis. Next, please. So we found that there is homogeneity among the group of developing new lesion, and we calculate disease severity based on several parameters: duration, number, and the surface of lesion. So we saw that the different parameters, presence of scars, ZCL scar, LST positivity, granzyme B superior to 2000 picogram per ml, indicate show an efficacy against the development of severe or intermediate form of ZCL. And the efficacy when combining leishmaniasis skin test positivity and production of granzyme B, we found an efficacy of 83.6% against protection against intermediate or severe form of ZCL. So next, please. To conclude, we showed a clinical and histologic differences between ZCL and SCL lesion due to L major, and we show a predominance of TH1 response within lesion. A significant difference was found between interferon gamma and IL-10 among this lesion. And interestingly, we show the presence of granzyme B and granulosate mRNA within this lesion, indicating the active process of parasite killing. And concerning the systemic immune response, we show the involvement of L major specific cytotoxic cellular immune response in host protection against leishmania infection. And we have to say that for old world CL, further analysis as needed to track down the role of cytotoxic human in the development or healing of the infection. Finally, the next slide, please. I would like to thank all members of our laboratory and other with whom I collaborate to this work. And thank you very much for attention. Next, please. And that's it. Thank you.
Thank you very much, Dr. Freya.
Sorry?
Thank you very much, Dr. Freya.
You're welcome. Thank you.
And now I would like to ask Professor Paul Kay to carry on with the session.
Okay, thanks very much, Khalil. So we've had a couple of great talks, and I'm sure we've got 2 more fantastic talks to come. We're running a little bit late, but I think that's not too much of a problem at the moment. So it's great to introduce the next speaker, an old friend and colleague, Mitali Chatterjee. Mitali is Professor of Pharmacology at the Institute of Postgraduate Medicine, Medical Education and Research in Kolkata. She's a pharmacologist, a medic, and has a PhD also from the Indian Institute of Chemical Biology. I think many of us in the field have known Mitali's work for many, many years, and her contributions to understanding drug responses, to understanding immunopathogenesis are many. And it's a great pleasure to introduce you, Mitali, and to hear what you're going to tell us about the immunology of PKDL and CL in Southeast Asia. Over to you, Mitali.
So thank you very much. Very kind of you for the introduction. And yes, it's been a wonderful collaboration over the years, and you'll see a lot of familiar information will be there, which we have done, which we have worked together. So good afternoon, everybody. I'd like to start with a brief introduction of the leishmania pathology, which we know is essentially when leishmania weds the macrophage. I call it a marriage of convenience because both need to survive and both do it very elegantly and graciously, if you might agree. So when the macrophages have the parasites within them, they essentially undergo alternate activation. And this alternate activation, in a way, is responsible for the immunopathology, because what it does is it impairs the capability of macrophages in terms of its antigen presenting capacity. It influences the T lymphocytes to move towards more of a Th2 response. And the fact that it's alternatively activated, it creates a milieu which is conducive for the parasite to survive. So they both live happily ever after, and that's how the disease actually continues. Next slide, please. So Colossal in South Asia, which is very similar to what Nikki presented, is essentially similar in that they present with fever and heterosplenomegaly. But what is interesting is the fact that they occur in a cyclical pattern. Is you have the disease, then you usually have a wave of intervention, which is usually DDT spraying. There is a dramatic drop in the numbers, but again, the numbers creep up. So we've had this, we've had these waxing and waning of numbers. And it's over the years that people have agreed upon the fact that possibly the South Asian variant of visceral leishmaniasis has an additional component, an additional layer of complexity, you could say, which is that of the dermal sequel to apparently cured visceral leishmaniasis patients, which is called post-Colossal dermal leishmaniasis. So post-Colossal dermal leishmaniasis is considered as the mobile disease reservoir. And the reason it's called so, next slide, please, is the dermal lesions that you see here, they're absolutely teeming with parasites. So the individual, whether the middle one, which is on the nose, whether you have them, they often occur on the face, they often occur in other parts of the body, but the dermal lesions being rich in parasites, the sandfly gets to take a rich blood meal, and these individuals then help to transmit the disease. But why I call them a mobile disease reservoir is that the lesions that they have, the treatment-seeking behavior that they have is something that they learn to live with the disease. They don't really think that it's necessary because it's not fatal. Unlike the visceral form, the disease is not fatal, and therefore there is no need to actually seek treatment. They are perfectly immunocompetent. They are, in every way they can just cover their lesions. So there's no reason for them to seek treatment. So it's important for us to address this component as part of the elimination program, because I think they are really the transmitters of the disease and we need to address them. So the next slide, please. So this is really what I'm going to be talking about, is the immunology of post-Colossal dermal leishmaniasis. And there's also an interesting twist in that Leishmania donovani not only causes Colossal and also causes PKDL, but is also responsible for a very fascinating variant, which is being seen in Sri Lanka. It's a cutaneous form of the disease. It's different. I will talk about the clinical features, and I will limit myself to cutaneous leishmaniasis in Sri Lanka, because the other form which is present in Asia is more closer to the old world, L major and L tropica, which has been recently covered. So I limit myself to Leishmania donovani causing parasites. So the Leishmania donovani parasites, which cause the two different forms of dermal leishmaniasis. So macrophages show a lot of plasticity, they show a lot of diversity, but what's important is they can either be the classical form where they will actually engulf a parasite or any microorganism and actually eliminate it. But here is a scenario where you have an intracellular pathogen. The intracellular pathogen needs to survive. So what it does is it undergoes alternate activation. And when it undergoes alternate activation, it actually acquires features, some of them which are important for this, as identifying features is CD163 being one of them, an increased synthesis of polyamines through an enhancement of arginase, increase in the IL-10 and TGF-beta, as also increase in the homing chemokines, which bring in the macrophages and the T cells towards the lesion. Okay, can we move on, please? So we first looked at the levels of cytokines within these cases, and what we found was, due to paucity of time, I'm not talking about the study population, but over a period of time, we've been doing this since 2005, and we have a repository of at least 250 to 300 cases of PKDL. So this was done through a bioplex assay where we looked at the cytokines within these PKDL cases, and this is at the circulatory level. So you can see it's a mixed profile of both Th1 dominant cytokines as also Th2 dominant cytokines. So what really we have seen over the years is that the PKDL cases show a mixed profile of a Th1, Th2 combined immune milieu. As far as the chemokines are concerned, next slide. As far as the chemokines are concerned, there is there's a decrease in the number of CCL21. There is a decrease in CCL21, which translates into a decrease in dendritic cells being homed. There is an increase in the monocytes. There is an increase in the T cells being homed, and there's also an increase in the B lymphocytes being homed. So there's predominantly the infiltrate that is there is primarily composed of monocyte macrophages, of T cells, and I'll talk about it. The T cells are mainly CD8 T cells, and you have B cells, both and a predominance of plasma cells. Next slide, please. Next one. Yeah. So the first thing that we needed to look at is there really an M2 kind of polarization, the alternate activation or the M2 polarization. So for that, what is an important feature is that there is an increase in the arginase activity. And the arginase activity has two advantages. One is that it synthesizes more of ornithine, which leads to more synthesis of polyamines, which helps in parasite survival. The other aspect is that by increased activity of arginase, you have a decreased activity of inducible nitric oxide synthase, and so you have less amount of nitric oxide being produced. So these are within circulating monocytes. We were able to show that at treatment, there was decreased levels of nitric oxide, which increased at treatment, as also there was a higher production of reactive oxygen species at presentation, which increased post-treatment. So the milieu that you have of these monocyte macrophages is essentially that which supports parasite survival, and so that's the one that we have, because you have more of arginase and you have less amount of nitric oxide being produced. Next slide, please. So we had an increased amount, as I said, the homing markers told us that there was increased amount of CD68 macrophages. There's a huge amount of infiltration which takes place. And what is interesting is with the 12 weeks of miltefosine that is given, there is a dramatic reduction which takes place. So these CD68 macrophages, next slide. These CD68 macrophages have several features of alternate activation, one of them being arginase. So this is CD68, which has been stained over here within the PKDL lesions. They expressed arginase, but arginase was also expressed in other cells beyond CD68. We still haven't delineated which cells are these. And when you can see that they're merged, there's a significant population of arginase, which is present predominantly in the CD68 dermal macrophages. Next slide, please. So there was arginase, which was elevated. There was also increased expression of CD163. This was CD163 within the circulating monocytes. CD14, CD61, 63, both together expressed a high amount of CD163. We also measured soluble CD163. I've not shown it here, but that was also increased. And we also showed at the dermal lesions, there is an increased mRNA expression of CD163. So it's two important take-home messages are, one is that PKDL is not restricted only to dermal lesions. There are also changes which take place in the systemic circulation. So it is a systemic immune response which takes place, as also that which is taking place at the local site. Next slide, please. So if you take it together, the important features that we have, which Nikki highlighted, is that whether it be visceral leishmaniasis or post-Colossal dermal leishmaniasis, is the milieu that is there is overall one of immune energy, either of exhaustion or of less presence of cells or of decreased antigen presentation. Altogether is basically producing an environment which is conducive for the parasites to survive. So you have the TGF-beta enhances the degree of polarization. It decreases the amount of dendritic cell migration. So there's less antigen presentation. The cells move towards more of a Th2 profile, and also there's an enhanced presence of Treg cells. So to see that what are the changes which took place in addition to the M2 polarization, we next moved on. Next slide, please. So we next moved on to looking at the dendritic cell population. And clearly, as compared to the healthy cells, there was a distinct decrease in the amount of dendritic cells, although there were a lot of cells which had infiltrated. The immune infiltrate is quite an overwhelming amount. This is a case of polymorphic PKDL where there's papules, nodules, and macules. But as far as post-treatment is concerned, there is a substantial reduction which takes place. But the dendritic cells and the dendritic cells also, to some extent, do return. So this helps in the impairment of the antigen presentation and is beneficial is beneficial to the parasite. The next aspect that we looked at, next slide, please, is a near total absence of CD4 T cells. This is very interesting because the previous speaker and also in the PKDL, which is seen in Sudan, there is a substantial presence of CD4 T cells, but we were unable to find any T cells, although there is a substantial presence of T cells. So to answer the question of what are the T cells which are then present, we looked at the status of CD8 cells. Next slide, please. And clearly, there was a huge overwhelming presence of CD8 T cells. There was a huge number of CD8 T cells which were present, which substantially decreased with treatment. We next wanted to identify that, like visceral leishmaniasis, where there is where these cells are anergic, is it a similar kind of scenario as far as the CD8 T cells are concerned at the lesional site? So for that, next slide, please. We went to look at the status of perforin and granzyme, and we examined the number of the cases, and in none of them could we actually find expression of perforin and granzyme. It did return. This is the positive control that we had, and we were unable to actually show any perforin or granzyme activity, which suggests that these lesional T cells, although they have perhaps lost their cytolytic activity, they are more, they have acquired the features of exhaustion, and that possibly helps. Even though they're present in large numbers, they don't actually, the disease, the chronicity of the disease still persists. Next slide, please. So if you take the whole story together, essentially what you have is in circulation, you have the M2 polarized monocytes. These M2 monocytes, because they have a less degree of proportion of CD86, and there's less CD28, which is present on the T cells, there is less degree of interaction between the cells. What is important is there is an increased presence of PD-1, which we know is a negative regulator of T cell activation. This was a very important finding, which is important as far as cutaneous leishmaniasis is concerned. They also had a lower amount of perforin and granzyme, and because of CCL17 and CCL22 being present, these T cells, CD8 T cells, were being homed to the dermal lesion, but in the dermal lesions, they were unable to perform. They had no functional role. because they didn't have perforin and granzyme. So parasite persistence continues because of this immunosuppressive milieu that has been generated. Next slide, please. So now, as we all know, that dermal leishmaniasis has many avatars, and I'm just focusing on one form, which is Leishmania donovani. And in Leishmania donovani itself, we have a very interesting variant, which is the cutaneous variant, which is not seen in India. It's not seen in India, Bangladesh, and Nepal, where PKDL is seen, but you have cutaneous leishmaniasis, which you see in Sri Lanka. And if you look at this profile that you have over here, the PKDL cases are sort of in between. They have both features of Th2 and Th1. And it's an open-ended question as to what is the scenario as far as cutaneous leishmaniasis in Sri Lanka is concerned. And I'll take you briefly through the data that is available regarding dermal leishmaniasis. Recently, Paul Kay's group has introduced a lot of interesting findings regarding cutaneous leishmaniasis in Sri Lanka, and I'll take that forward. Can I go forward, please? I think I'm running a little short of time. If I'm given 2 more minutes, I'll quickly run through it.
If you can, please, thank you.
Okay, so cutaneous leishmaniasis in Sri Lanka. If I can go on to the next slide, I can actually I can skip this. So what's important is, I think is important is that it has several similarities, but it also has important differences. Considering the homogeneity of Leishmania donovani, which is, they're both MON37 as far as their zymodyne is concerned. They are both transmitted by the sandfly, but as far as their lesions are concerned, PKDL is multiple lesions. PKDL is a systemic disease, whereas cutaneous lesion is a single lesion and is not a systemic disease. They also differ in their treatment-seeking behavior. Usually the disease duration is not more than 6 months. I'm sorry, this should actually be less than 6 months. And so we don't know whether they're actually self-healing. At the moment, they receive sodium antimony gluconate. So they're of a shorter duration. They are usually single lesions. And what's important is they're also ulcerative lesions. And another very important finding is they're quite resistant to treatment, unlike PKDL. So in terms of the histopathological changes, there are a lot of similarities. They have a considerable amount of immune cell infiltrate which occurs. As far as the morphological changes are concerned, they're very, very similar. There is the same degree, the epidermal atrophy, the follicular plugging, the hyperplasia. They're both very comparable. One important difference, which is notable, is the fact that you don't have a granuloma within a PKDL, which is why it probably can be diffuse in character, whereas with the Sri Lankan variant, the granuloma is present, which probably helps to contain the disease. Next slide, please. In terms of the immunological responses, there is not a lot of work which has been done. It's only very recently that work has emerged, but comparable is the fact that both of them have alternatively activated. One has more of alternatively activated macrophages, whereas here they tend to be more of classically activated macrophages. They both have a Th1, Th2 milieu because both produce IL-5 gamma as well as TNF alpha, IL-6, as also IL-4 and IL-13. Here there is a much more of CD8s which are present than CD4. We don't know the scenario as far as Sri Lankan variants are concerned. Next slide, please. And this is data which is really fresh from the oven, and where this is spatial profiling and fluorescent in-Situ hybridization, which was done. So work which was done in collaboration with the group of Paul Kay and the group of Shalindra Ranasinghe in Sri Lanka. And they examined cases of cutaneous leishmaniasis before treatment and after treatment. And the take-home message they said that they did present was that there is a reduced expression of PDL1 and a reduced expression of IDO1, which is indolamine 2,3 dioxygenase. And both of them are very important both of them are important inhibitors of immune activation. So their reduced expression suggests that with treatment, there is immune activation which takes place. And they also strongly suggest testing of PDL1 inhibitors, which can be done in all forms of leishmaniasis, perhaps. So this is really, I expect that more data will emerge from this collaboration because they have access, they have done it in terms of spatial profiling in that regions of interest were selected. And they've also done it at a genetic level to look at the expression of various immune genes which are related to cell function, and as also in both in terms of T cells as also the other cells. Next slide, please. So this is really the story that I tried to put together. Time was rather short, but I hope I have been able to convey the overall scenario that is there in PKDL, which has similarities as also differences with the cutaneous leishmaniasis that we see in Sri Lanka. This is the team. There is a female predominance. The disease doesn't have a female predominance, but my researchers do. We all passionately work towards potential elimination of PKDL and also help in knowing more about the immunopathology of both types of leishmaniasis. Thank you very much for your attention.
Okay, great. Thank you very much. That was fantastic, Mitali. Can I just please remind all the speakers and as well as all the audience to look at the Q&A, to put questions in there, please, and they'll be answered live by the speakers, and that will help us keep a fairly short Q&A session right at the end of the talks. So please do check the Q&A and put your questions in there. So I think when Professor Khalil introduced this session, he mentioned the importance and the early work on vaccine development. So I think it's fitting that we end this session with really an update on one of the lead candidate vaccines for leishmaniasis currently. So it's a great pleasure to introduce Dr. Selva Pandian. Selva is a PhD training at the International Center for Genetic and Engineering and Biotechnology in New Delhi. He spent time at the US FDA for his postdoctoral work, where he was really instrumental in establishing the role of centrin in parasite division and really setting the scene for developing what he's going to be telling us about today, which is the development of a live attenuated Leishmania parasite as a vaccine candidate for VL. So it's a great pleasure to welcome you, Selva, and to hear what you're going to tell us about the latest in this exciting development.
Yeah, so thanks, Paul, and then the organizers for giving me this invitation here. So may I share my slides? Please allow me.
Please go ahead.
Yeah, please allow me to share my slides, my screen. I'm presenting my slides myself, so please allow me to share.
Can you get that at the bottom of your screen, Selva, the share button?
Yeah, the host disabled, so they have to allow me. The host disabled participant screen is sharing. So I'm waiting. Yeah.
I think they will. Sorry about that. They will connect you shortly. Yeah.
Okay. Yeah.
Right.
Fine.
But we should be able to share now.
Right. Now, do you see my screen?
We can, Selva. Thank you.
Very good. Thank you. So, so I will be talking on the development of mainly centrin-deleted live parasite vaccine for VL. In the process, I'll also give introduction on the prevailing other vaccines and the development scenarios. So the reason for vaccine is that in general, the perception that the medication, the drugs, they have severity, toxicity, and drug resistance is a problem. So vaccine definitely will work based on the concept that we have several diseases, infections all around us, and bacterial infections, viral infections, and many vaccines, either they are subunit based or killed or live vaccines. They're all in practice. So why can't we have something like that, one type or the other for leishmania? That's the concept. So the best way to escape, obviously, is having vaccine. Right. So yeah. So 4 canine vaccines are commercially in use, like Leishimmune, Leishtec, Canilaish, Letifend. And I will not go into the detail of what kind of formulations. They're all here, and then the percent efficacy is all here. Leishimmune, for that matter, from 2014, it was withdrawn based on the few adverse reactions, and then also efficacy related issue. Otherwise, there are other 3 vaccines for dogs are available, but the question is, why can't we have them for use in human? What I'm hearing from people via communications is that either the market for dogs are more than human, and then also for human, most probably what I feel that the regulatory the aspect, the screening, that the demand is probably more, so it is limiting that towards coming to the human side. But however, I wish it comes out sometime, we have successful vaccines even for humans. So this slide shows, is compiled by Dr. Paul. It shows a few current vaccines that are in development phase. However, this little old, a couple of years old. Since then, what we see is that Mulibaclin and then Mologian. These 2 have been, withdrawn either due to issues or fund related. However, the other one, it's a live attenuated vaccine by Leishmania major syndrome-related parasite. I will come with that some information in subsequent slides of mine. And I would like to specifically mention Dr. Pal's CHAD63KH. That's doing well in terms of clinical studies. They are doing it with Wellcome Trust Fund in Sudan. And if you can see that it's mainly they're trying to have it like therapeutic vaccine for both VL and PKDL. And they are having considerable progress in that. When they looked at very briefly, if you look at this graph, so 42 to 90 days old, they looked at the percentage in disease over time. So with some certain people, patients, they see 25% clinical improvement, and with some they see from as high as 60 to 90%. So, but for yet different number of people, they get cured completely without even having a chemotherapy. That's something really remarkable. So I wish that group, Dr. Pal's group, and then with the collaborators in Sudan, they'll have a major success soon. And then if you're coming to the other aspect of live attenuated vaccine against VL, this was when I joined US FDA in 2000 in Dr. Hiran Akashi's lab. And I stayed there for 10 years as a regulatory officer as well. So in that process, I was instrumental in developing Leishmania donovani deleted for a growth regulating gene, cell division specific gene, centrin. When that gene was deleted from the parasite Leishmania donovani, it is specifically attenuated. The growth was attenuated in the emasticode stage, not in the promasticode stage. As you can see from this, that the blue lines signifies the flattened growth in the emastigodes. However, the growth is not that much affected in the promastigodes. If you look at a little bit more in detail what is happening with these type of gene-related emastigodes, there is a nuclear division which is not affected. So instead of one nucleus per parasite cell in emastigodes, there are multinuclear condition. That means nuclear division is keep happening. However, cell cytokinesis is not happening. So this particular gene centrin is associated to basal bodies. From the other angle of electron microscopic images, we can clearly see that there is a problem in the division of basal bodies. Okay, that's the reason probably the cell cytokinesis is not happening. So anyway, we see a huge ball of emastigodes at the end, and this is in the in vitro condition. Similarly, in the ex vivo condition, in the macrophages, we can see the multinucleated huge ball of emastigodes, whereas at the left you can see it in the control, the wild type parasites growing happily. So the bottom is the graph representing that data. And further, we have tested for its safety and efficacy in mice, hamsters, and dogs. This clip shows its immune responses towards Th1 in dogs when it was tested with our collaborator Ricardo Fujiwara in Brazil. So he compared with the leishmaniasis. There is a dog vaccine. Of course, this was before the leishmaniasis was withdrawn. So we worked like 2012 and 13. So compared to that, as an antigen, the whole parasite, Leishmania syndrome parasite, works much better in terms of improving the Th1 response. And what we see here at the bottom is the parasite number and correlating to that. And then we also tested for its recall response in human PBMCs coming from PKDL and healed VL patients. Okay, so in that case, again, what we see that is that like you have the PBMC isolated from these people, expose it with live parasites, and then look for the immune responses. What we see clear increased Th1 response, IFN gamma, and then not so much increase in the IL-10. This we see only clearly in the either of the case. That the people who have already encountered the parasite in their body, like PKDL or healed visceral Ishmaniasis patients. Otherwise, in the control, we do not see this recall response. So clearly signifies that there is a protective immunity also happening in these cases. So this is what is the whole nutshell about the Donovani centrin knockout parasite. And we have tested for its cross-reactivity against challenge with the CL causing parasite, major, as well as in fact, even to Braziliensis. And then we recently worked with the preclinical toxicity study in animals in Vimta, Hyderabad, with a fund from BIRAC, and that is complete. It's almost complete and giving no toxicities, and that manuscript is currently in progress. So on the other side, and again, see the last bullet in that for human, however, safety is still a major concern. There are a lot of discussions happening around that whenever we present, they always come out and say because it's Leishmania Donovani, and it's again, there is a major concern if you are going towards regulatory aspects for clinical trials also. So on that background, Dr. Nakasi with other colleagues, peers, Dr. Abhay Satoskar from Ohio and Greg Metleski from Canada, they worked out similar gene deletion in Leishmania major parasite. If it similarly happens as an attenuation, maybe this can be a better option than Donovani. So they have came up with a knockout parasite via CRISPR based deletion, and they've succeeded, and they've tested for its immunity in mice and hamsters against CL and VL causing parasites, and they've even tested needle based challenge and then sandfly based challenge, and then they looked at the immune response in detail. So they see the protection similarly against both CL, that's homologous, and then heterologous challenge with VL. So they see protection. And then in the nutshell, few clips, I would say that in that work, they have tested Leishmania major, centrin-related parasite, and when they immunize and challenge in the ear, they don't see inflammation and lesion, whereas the naive animals, if they are challenged or non-immunized challenge, and they see the clear disease symptom, and then they don't see the parasites in the immune challenged animals. This is the case in the left side. What you see needle challenge based, or in the right side you see sandfly based challenge. And then they came out with data, huge amount of immune responses, and I would not get into that detail, all correlating to Th1 immunity. Then they also, to see whether it protects against VL, so immunization with the major parasite, and then challenging with the Donovani. What you see here is the hamster immunized, showing normal spleen, healthy enough compared to the other one, non-immunized, challenged, having huge spleen and then retarded growth. And then you see there is a reduction in the death. I mean, there is increase in the death of the non-immunized challenge, whereas the immune challenged are healthy enough. So, and back in our lab, we are working with the centrin knockout parasite, centrin deleted Donovani, which we have, and we looked at, we are rather looking at the proteomics profiles in the parasite as well as in the host after infection. That thing is going on, and parallelly, we also got inspired that we should also do similarly some gene deletions in the cutaneous causing parasites. So one thing is we thought, let's start with Leishmania Donovani centrin 4. This parasite has 5 different centrins. We thought we'll take the other centrin 4, which seems to be expressing more in the imastigote stage. So we confirmed that, and then we deleted this gene, centrin-4, in Leishmania mexicana. We couldn't do the deletion in Donovani. Probably it is essential for that parasite, but for mexicana, we could delete centrin-4. And then we see the attenuated growth in promastigores as well as emastigores. Okay. However, when we wanted to check their replication in the macrophage, we found that this parasite is not attenuated anymore. So that was the end of the story at that point. However, we did a lot of characterization on the gene part, and the manuscript is in the process actually. So we came out with another such gene, adenosine kinase like protein, that is also emastigore specifically expressing 31 kilodalton, and it is in the purine salvage pathway. So we deleted successfully by CRISPR in Leishmania mexicana. Interestingly, what we see is that the promastigores do not have no flagella after deletion of AKLP gene. Okay, we confirmed through even EM studies. The question is, we would like to see the flagella biogenesis with this kind of mutations, and also the fate of AKLP knockout emastigores, whether it can be tested further as attenuated vaccine. So the preliminary studies, we try to see its growth. It's attenuated in the emastigore stage. Probably flagella is something important in many aspects in order to initiate an infection. So this is the thing with in vitro. However, when we try to check in the macrophages, we do not see its growth in the macrophages as compared to wild type cells. Okay, with this, and we are currently in the process of testing further in animals with our JNU collaborator, Dr. Nitipuri. This is with a grant from Department of Biotechnology. So the conclusions, I would say that emastigore specific genes you can trust on to characterize, to know more on the biology, why they are needed for emastigore stage. That's one thing. And also, if you are successful in getting an attenuation in that particular stage, you can easily, obviously, test for its vaccine efficacy in the animals. Then, so more research efforts are needed to delete such genes. If they're essential genes, there is always a big task to delete such genes. So maybe one can try with the conditional based knockout procedures or dickery based procedures. That's something recently people have tried in other parasites. So our lab with efforts are continuing towards such optimizations in the CRISPR gene deletions. So I would like to go with acknowledgement. My students, Kavita, Rati, Adil, Ankit, Roshan, Enam, and Noor are very much actively working in different aspects. My collaborator, Dr. Nitipuri, JNU, will look out who takes care of the animal studies, and then Yuan Stekers from France. He is guiding us towards the gene deletions using CRISPR. And then, of course, I'm still in touch with Dr. Hiran Akasi from US FDA for his inspirations. And then we do collaborate with certain specific aspects, even with the Donovani, knockout parasite. So I'm ending my conversation. So thanks to the organizers, Dr. Paul, panelists, and the audience.
Great, thank you very much, Selva, for keeping time as well. So we've got 10 minutes left. That was a great presentation. And I think it's also not only good to hear about the progress being made with the centrin knockouts, but also to hear that you haven't stopped. You're still thinking about other alternatives. And I think that's a really important message. We have a very small number of vaccine candidates in clinical trial. We need the discovery science to continue, but we also need to open up that pathway to allow more of those discoveries to get into early phase studies. And I think that's going to be essential to make progress over the next few years. So I think we've got time for a few questions. The first one is from Charlie Mowbray to Nick. And the question is, you mentioned that a higher proportion of relapsers were young males working in the fields. So are you sure that these are relapsers rather than reinfection? I understand that migrant field workers are a higher risk group for infection. Nick, do you want to address that?
Yeah, sure, definitely. Yeah, there's multiple components to this question, or at least to the answer. The easiest would be to reference quite a recent paper where they show in quite a similar group. It's actually in Ethiopia as well, in the same region, that it's really relapses and not reinfections. But of course, it might be a little bit too easy to reference to another cohort study. One reason we suspected it was reinfection, relapse instead of reinfection, is also because it happened quite soon and quite frequently. And the proportion of field workers was not different between the patients that did not relapse and the patients that did relapse. So that already gives us kind of a suspicion that it's rather relapse than reinfection. But in another sense, it does not matter that much for what we are trying to do, because we are trying to predict susceptibility for relapse, but it could also be for reinfection, because there's a subset of patients that do not develop this reinfection if they're the same susceptible population for sandfly bites, but they do not develop a reinfection. That also means that they have protective immunity probably towards infection. So we are not, in that sense, we would be predicting susceptibility for VL symptoms in general, and not necessarily relapses.
Okay, thanks very much. So a couple of questions to Selva from Fabiana Alves. Do you foresee this vaccine mainly for prophylaxis? This is the centrin knockout for prophylaxis and or also for therapeutic, for example, in PKDL patients? And can you just update us on the progress towards phase 1 trials for the L major centrin knockout? And I guess that relates also to a question from Mitali about whether we should be really looking at therapeutic or prophylactic vaccines.
Okay, so what I know so far on the clinical aspect of Leishmania major centrin deleted parasites, they are about to, they are probably onto the toxicity analysis in the animals, or probably it's over, and they're probably switching over to clinical studies with a GIT fund. That's one thing. And then it's currently speculated that as a prophylactic vaccine. However, Dr. Nakasi is also trying and towards therapeutic. They're also working and they're doing some studies in that aspect also. Otherwise, majorly we have been thinking all these along like prophylactic only.
Okay, thanks very much. Maybe I could do just a little bit of self-publicity in relation to the question of therapeutic versus prophylactic. There are scientific considerations, certainly, but there are also considerations related to manufacturing and demand, which would actually allow the development of those different forms of vaccines. And if anyone is interested, we published recently in PLOS NTD the first analysis of the global demand, likely global demand for different forms of vaccines for leishmaniasis. So I just point you in that direction. Do we have any other questions from anybody? I don't see any others coming up. I'd be quite just a point of interest about Thuria's presentation. It's really great to see in Leishmania major that you're starting to look at human disease. I think Leishmania major, even with its incidence, has always been dominated by studies in the mouse. So it's really nice to start to see some of this data emerging from your clinical studies, and the prospective studies are really important. The reinfection, it really reminds me of early studies in the mouse work from Ingrid Müller and others that also suggested that CD8 T cells were predominantly important for protection against reinfection, which obviously has a knock-on effect for thinking about mechanisms of vaccine-induced protection. So just congratulate you on that study, Thuria. It was really, really nice piece of work. Don't see any other questions.
I have one question.
Yes, Selva.
Yeah, so this is regarding So next presentation on the relapse of the leishmaniasis in the HIV patients, also with Dr. Mitali on the PKDL, okay, emergence. So in either case, I'm just thinking towards CDs and other molecule exhaustion, those cells, exhaustion of those cells. Do you think the reason could be prolonged antigen stimulation during chronic infection? So the parasite we think that the parasite is gone once again it is relapses. In either case, PKDL also the VL is gone, but maybe there are some parasites still there chronically, and that comes out. That's also like reason for exhaustion. I would see. I never worked out myself. So
do you want to address that, Mitali? This question of whether there are
sure. I'll be happy. Anyone, yeah.
Subclinical parasite burdens after cure.
Mitali, I think you're on mute.
That's a very relevant question that you ask. And it's important because we really don't know whether there are parasites left in the dermal macrophages, because the problem actually is that the gap between the person having VL getting cured and the person having PKDL appearing, there's a gap of at least 2 years, minimum of 1 to 2 years. So if we could follow up the cases and if we could see whether there were similarities, because I think this is a question that a lot of people wanting to ask is the PKDL parasite different from that of the VL causing parasite? Because if you look at the Sri Lankan variant, here is Leishmania donovani causing a localized cutaneous lesion. Here is another donovani causing a systemic lesion. So that would really be interesting, and I think that can be answered if researchers in Sudan and in East Africa look at isolates from cases of VL and look at cases and look at the same case which develops PKDL and then compare the two. That would be an interesting follow-up. The second part was, I think it's more to do with impairment of antigen presentation, because the monocytes are they're becoming alternatively activated. So it's more of impairment of antigen presentation. So from that, it basically makes the CD8 T cells just go to sleep essentially. So that's how they do.
I think we've got 2 more questions. I'm just going to have to move it on. A quick comment from Thuria, I think, and then I'd like to hand back to Professor Khalil to either make a comment or question or close the session formally. Thuria, quickly, please.
Okay, thank you. Just I want to say thanks to Professor Silva, and I have just a comment that concerning the L major centrine. We are doing collaboration with Professor Hira and Abaye, and we are testing the immunogenicity and efficacy of this vaccine within dogs. So that's it. And it's ongoing work.
That's really good to hear. Good to hear. We look forward to the results from those studies. Professor Khalil, back to you.
Thank you very much, Professor. Okay. I would like to thank the presenters for this basic information that we got about L major and infantile disease and intralesional immune responses that may be helpful in the future to develop vaccine or to modulate vaccines. And I don't think I have any more thing to say, but to thank Professor Kay and to thank the presenters.
Okay, so thank you all the presenters, everyone for listening, and I think we can formally close the session at that stage.
Thank you very much.
Thank you.
Thank you. Thank you, everyone.
Thank you very much.
Back to you, Monique.
You're welcome. Thank you very much. So I think we'll be ready then to move to the next session. Please don't go away. We're moving into the next section. Okay.
Do we have 5 minutes for stretching our legs?
Yes.
No, just
Good afternoon, everybody.
wait,
Stretch
wait.
your legs. Hello.
Yelem is here.
Yeah.
Just maybe to stand.
Good afternoon. Are you ready to go with us? Ready?
Yeah, 5 minutes, Yelem.
Okay. Thank you very much. I'm Yalem Tsahai Makonnen from the College of Natural and Computational Sciences at Addis Ababa University. I'm the founder of, one of the founders of LEAP. This session is a very relevant session. It's not to belittle the other sessions, but the impact of COVID-19 on NTDs and training on fundraising tools in Africa. And my co-chair, I want to briefly introduce the co-chair, Dr. Erin Mukhiv. Dr. Erin Mukhiv joined DNDi in March 2020 as HIV Access and Medical Affairs Leader in the DNDi Africa Regional Office. She is a medical epidemiologist with over 17 years experience in clinical and health program management. She holds a Bachelor of Medicine and Bachelor of Surgery degree from the University of Nairobi and an MA PhD in epidemiology from the University of Washington in Seattle, USA. Dr. Mukhiv has been involved in implementation research across HIV prevention and treatment, with her current focus being on pediatric HIV treatment and outcomes, mental health among HIV-infected adolescents, and HIV drug resistance. So I want to remind the participants that this conference is live streamed in hashtag leishmaniasis, hashtag Africaidea, and hashtag fit and TDS. And some other reminder again, presenters, oh my God. Okay, I thought the internet interrupted. Okay, do you hear me?
Yes.
Okay, so sorry. Presenters are encouraged to have their cameras on, and all other microphones will be muted throughout the presentations. And again, the live Zoom session will be recorded for those who are not able to attend. So if you don't want to be included in the recording, simply turn off the camera and the microphone. Questions and answers, as usual, as on Q&A, and also the chat function can be utilized for any other interventions. Now let me go directly to a very brief introduction of the first speakers and the training aspect, which we are going to have for 1 hour. We're excited to have it. And the trainers are Karin Genoka Services President, a graduate of the London School of Economics, United Kingdom, and of a leading management school in France. She has more than 20 years experience in international fundraising as head of fundraising at the Drugs for Neglected Diseases Initiative. Karin helped raise 130 million euros for medical research. She's also a president and senior consultant at Genoa Services. As part of her daily work, Karin identifies funding opportunities tailored to projects and organizations, guiding them in their relationship with funders and developing grant application with their teams. And the second trainer is Bineta Ba. Is she around? Bineta Ba is an expert in sustainable development and program management. She has 20 years of experience working in Africa, America, and Europe, particularly in Switzerland, where she has worked on large projects, collaborating with major public and private funders and United Nations agencies. So she is the founder and managing director of BFK International and the Forum of Technological Innovations for Energy Transition, Environmental Safeguarding, and Sustainable Development. So now it's my pleasure to invite the presenters, and hopefully we'll have 1 hour of a good ride in the training on fundraising. Thank you very much. The floor is yours.
Thank you, Professor Mekonnen. Thank you, Dr. Mukhiv. for inviting Binetaba and myself today at the LEAP conference. And we would like to welcome everyone to this session on key tools to raise funds for projects on organizations in Africa. So next slide. So here you have a summary of the profile that has been given, that has been presented. We won't go through our profile again. I'll just give some introduction words before the training. And I'd like to give you 3 reasons why fundraising should be on the top of your to-do list in the coming days and months. And those are the 3 reasons. First, we all have faced the fact that funders tended to prefer funding COVID projects for the last, since the beginning of the pandemic. But this situation has been changing for the last month. And there's a space to get some interest from funders regarding leishmaniasis projects at the moment, in a context where funders have an increased understanding of infectious disease and health research. So for that very first reason, I would encourage you to really consider fundraising as your daily task. The second reason is it has to be, there's a tendency amongst funders to look for giving direct funding to local organizations. Oops. Can you hear me?
Yes, we can hear you.
Okay, sorry, there was an alarm. So the second reason is that funders want to provide funding to local organizations. And today is the forum where lots of you are close to the problem you want to solve. And then you have an excellent pitch to raise interest amongst donors about your projects. So again, second reason why you should consider doing some fundraising as of the end of this conference. The last reason about the importance of fundraising is I've been observing, I've done fundraising for 15 years, is that there's more and more very useful information available on the web that you can use to really guide your fundraising effort. And we, together with Binetaba, going to show you today some useful source of information or tools you can use freely to guide your effort and be successful with securing funds for your project or your organization. Okay, so Binetaba, could you continue? Thanks.
Thank you very much, Erin, for this introduction. I would like first to thank a lot the organizers of this live webinar. We are very grateful to be invited here, and it's a great opportunity for us to share with you some tools and some guidelines for successful fundraising. As Erin said, I will not add a lot in this introduction, just wanted to say that this is like a whole training course that we deliver in 5 days chrono. And for this purpose today, we are just summarizing it for this course to fit in 1 hour. So we'll do our best to give you the most important tools, techniques, and knowledge that can be useful for you and for your project. So to to introduce the key tools to raise funds for projects and organizations in Africa. Here is like the 4 points that we would like to develop in this presentation. So after this presentation, we hope you will be able to find clear answers to these 5 points. So the first one we are going to talk about is what are the different types of funding for your organization or project? This is very important question because many organizations, many individuals, when they have a project, this is usually the first question that they ask to themselves. Where am I going to look for fundings and what types of fundings are available for me? The second question is, how can I identify the donors who provide funds in my country and my sector? This is a very important question also because you know that donors will donate according to their priorities. So when you have a project, you need to identify the donors that fund the type of project you have and the donate that fund in your country and your sector. So this is very important. The third question is, where can I search for calls for proposal? This also is very important because when you have a project, you look for types of fundings, you look for donors, but where will you go to look for those information? Probably you will need some platforms, some maybe to go to some events, or we'll talk about all these places that you can go to look for call for proposals. Yeah, where to go to search for those calls for proposals. The fourth question is, how can I increase my chance to be of being funded? So to answer to this question, we're gonna go through some tools, some techniques, and share with you different type of things you can do to increase your chance, because there are a lot of fundings, but it's demanding and it's competitive. So we'll try to look at this question. And the 5 and last is the 5 steps to start fundraising for your organization today. The whole process you should go through for a successful fundraising. So the next slide, what are the different types of funding for your organization or project? In this slide, so we see that you can have different types of fundings, institutional fundings, including the financial institutions, the development bank, like the World Bank, like the African Development Bank. I will just ask the people, the one who is managing the slide, to go through and to show the next sections exactly. You can go through until you show everything. Thank you so much. Yes, the previous one. Excellent, thank you. So here, the different types. So when you have a project, what you should know is that you have different types of fundings. You should know those types of fundings, and if they apply to you, if these fundings apply to the type of project that you have. I was talking about institutional fundings, including the development banks like World Bank, African Development Bank, the Islamic Bank, etc. Those are the institutional funding. And also the multilateral or regional fundings, including European Union, and the national fundings, including like, for example, the government of Canada, USAID, etc. All the fundings coming from developing countries. You also can have some grants, fundings from large foundations like the Bill and Melinda Gates Foundation that is funding medical research project. You have the MasterCard Foundation, which is in the category of the large foundations, and you have small other foundations, hundreds and hundreds of foundations. So you can also look for fundings in funding coming from individuals, from higher net worth individuals. You can do crowdfunding, which is like very common today. A lot of crowdfunding platform online. You have the legacies, and you can have also funding from companies, private companies, that have like corporate social responsibility department that are giving some fundings. You also can have fundings from your sale, your product services, etc. But I think that for the purpose of this live webinar, we can focus on the institutional fundings from multilateral, regional funding, national funding, the funding from development banks, and from foundations. These are major donors. We can go to the next slide. So for the COVID-19, you can go ahead. You can show everything in this slide. Next, great, that's okay. Okay, so here is like a picture of the COVID-19 funding, so that could be very interesting for you to know. If we go to the institutions and the funders, you can see that the major funders of the COVID-19 are governments and multilateral donors. And if we go to the left side of this slide, you will see that in 2020, government together gave up to $12 billion in terms of fundings of projects related to COVID-19. The International Monetary Fund gave up to $4 billion, etc. The European Union also is a major donor for COVID-19. So all those fundings aimed to fund like activities, program, grant, contract, etc. And you can see that how the fundings are ventilated throughout the world, majorly in Africa, in Latin and Central America, in etc. So this is very important for people working in project related to COVID-19 to see how like the funding, where the funding comes from, and the type of support they can get, and wherever they are in the world, they can see who is funding a project in their area or country. You can go to the next slide. Okay, so it's a long road for funders to decide to fund a project. So, and it's important for any person or organization having a project to fund to know this process. So usually donors, they have priorities, their own priority. They set their budget, and if you know the donor's priority, it's pretty easy for you to see if those priorities align with your project and your organization priorities. And this donor will be a potential donor for you. Then the donors will go, will release a call for proposal. They may do so, they may not. And then they go to the third step, submission and review of proposal. They will check like eligibility of your project. They do all the necessary reviews, and then if like you have like you succeed, you will go to the next step, which is signing the contract. and then the reporting and the closure, etc. So in that last step, it's very important that everything you decide or you committed in this project, that you do it, it imply like all the reporting, the monitoring, audits, etc., and the whole implementation of the project until the project closure. So this is usually the process that funders are going through, from the funding priority, the budget setting, to the contract and implementation of project reporting and closure. So if you go to the next slide, I will give the floor to Erin to continue. Thank you.
Thank you, Bineta. So now that we've seen that there are many funders, potential funders that could support you, an important question is, how can I identify the funders for my sector who share the funding priorities as described by Bineta that are aligned with what I'm doing? In your area, you are very lucky. In fact, you can consult a very useful report called the GFinder report. This report compiles information about R&D for neglected diseases, including leishmaniasis. This report is issued once a year, but what is very useful, what is available online for you, is the data they use to write this report. So for the purpose of this conference, I went to this website on the top right, and I filtered the information on leishmaniasis. And there, in just a few minutes, you can learn how many, an average of 40 million USD is spent for research on leishmaniasis over the last few years, that the 3/4 of this funding is disbursed by public donors, so the national donors Bineta was talking about. And you can also learn, digging into this information, that the main funders for leishmaniasis R&D are the US, the UK, Brazil, and Canada in terms of national funding. So very, very useful information in this report, but before all in the database that is available for you to consult on your area of focus. Okay, so those are data about who funds global R&D for leishmaniasis. Next slide. Another important piece of information is, okay, this is the global picture, but who in my country, where I am located, where I'm working, who gives money to this country in my area? And here I would like to invite you to use the Sdg Financing Lab tool created by the OECD. It's a very, very fun tool, actually. You go there on Sdg Financing Lab. You can do it while I'm talking. You can type that name in your browser. You will arrive to this page, the Sdg Financing Lab. And there you can filter the information about funding by either selecting providers or recipients. So for the purpose of today, I clicked on recipients on the left. I entered Kenya, thanking the host here of today's conference. And I selected, so this is a database of funding for the SDGs. And of course, because we all are working on health, what is interesting to know is who gives money to Kenya for SDG 3, which is good health and well-being. On this tool, Sdg Financing Lab, when you enter this information, shows a globe, like on the middle of the screen, with the different flow of money that goes to Kenya to support SDG 3. Okay, so it's really a fun tool to look at. If you click Explore on the top line, on the top of your screen, you can dig further this information, and I extracted a piece of that following screen on the right. You can see that the main funders for SDG 3, so health in Kenya, are the United States, a bit like what was highlighted in the GFinder report. And key funders in Kenya for health are the Global Fund, which actually don't give money for leishmaniasis. So then you need to investigate their funding priorities. The UK, UNICEF, and Germany. So thanks to those two tools, the GFinder report and the SDG Lab, I narrowed down, the level of the many possibilities about funding to those who are interested in your area of work and your country. So this will help a lot to decide to check for calls for proposals coming from those institutions, or build a relationship with them to propose your projects in an unsolicited way. Okay, next slide. Next. Okay, the third question, now that we know there are lots of funding, we know where to look at to identify my prospective donors. The next step is to know where to look for calls for proposals. And you can check the websites of the funders we have identified, and we'll look at that after this slide. But here I'd like to highlight that you can go to portals such as DevEx or Funds for NGOs, which gather for you all the information about the coming call for proposals. Like you have many calls for proposals covering many subjects, many areas, many types of organizations. So those portals allow you to filter the information that is most relevant to your area, and screen down, select call for proposals that are open and that you can consider to apply to. Of course, once you identify those calls, you need to clearly check if your organization is eligible. For example, a call for proposal can be designed for institutions in Kenya or in the region, but not somewhere else. So you really need to look at each call and check if you're eligible to submit a proposal. Next slide. So you can check for funding opportunities on those generic portals like DevEx or Funds for NGO, but you can also go to the websites of the funding prospects you have identified. So for example, the NIH has a very good website where all the calls that are coming, the funding opportunities, active funding opportunities are listed. So when I created this slide, there were 15,000 of them. So of course, there's a lot of possibility here to filter the information to check which ones are more relevant for you. So that's the NIH in the US. Next slide. If you want to apply for funds to welcome, again, you have available information on their websites on the type of grant schemes they offer to support projects. So again, go to this website, check if you're eligible, if it's in line with your project, with your organization. Next slide. We saw in the GFinder that Brazil was a big player in terms of leishmaniasis, obviously. And here you have on the FAPESP website the funding opportunities you can apply to. So again, check the website. All the information is there. Lastly, last example, you can go on the EU website. So as for the NIH, there are many calls for proposals. So again, here you need to take time to find your way about filtering the information to select the most relevant calls for you. So for example, here it's the EDCTP project. Okay, so that was about selecting a call for proposals. And now we're moving to the stage, once you have selected your calls, once you have selected your prospective donors, the next step is to submit, to write a proposal, and I give the floor to Binetta.
Thank you, Kevin. So now you have your prospective donors and you want to start writing the proposal. So you will need to know what to do in order to increase your chance to get funded. So first point is you need to know and understand your prospective funder, who the funder is, and to know the priorities of the funding. I know at this step you already checked that you are eligible, but it's important to align with who the donor is and to understand everything that the donor wants to see in your proposal. The second point is you need to present the project with the right level of detail. We know that we are in research, medical research project, scientific field, and sometimes a lot of scientific information. But at that very moment, it's important to get closer that what the donor needs in terms of detail and to give that level of detail. Not a lot of scientific detail, not too much, but just what is very important for the donor to understand your project. So a very good balance between all the information you have, the scientific information, and what is needed by your funder. Accurate information. And in the third point, you need to build the trust with the donors by asking the right question. It's an exercise of communication in this step. Communication is key. Do not hesitate to ask a question, to write emails, or if you have telephone, to give phone calls. And most of the time, you will have a contact person to the funder and use that contact person to communicate and to ask the right questions that will give value to your proposal and increase your chance. So for the next step, we are going to talk about some tools you can use for your proposal. I will introduce the tools, and in the next coming slides, we'll go through those tools. The first one is a typical proposal template. You can go ahead, yeah. Most of the time you can ask yourself, oh, what should I put in my proposal? What should I write? What should I talk about? But if you follow these template, you are good to go. So in your proposal, you can put these 8 points. First of all, the description of the context, the current situation in which your project is, to describe it, Is it a pandemic context, epidemiology context? You will know. So a very good and accurate description of the context. Talk about the objective of your project, what you are aiming to do with this project, and the overall goal and the overall project of the overall purpose of your project. You can talk about the beneficiaries, also who they are, for whom like the project is designed. So the first point you can talk about in your proposal is the implementation plan in a very clear way that will help you to reach the objective that you have described in point number 2. Also talk about the expected results and income after the implementation of this project. Present your organization and your team. It's a very, very important point. You would like to show to the donor that you have the good team to implement this project, that your team have the expertise, the necessary expertise to implement the project. If you have partners also, this is the moment to talk about your partners, their role in the project and their expertise too. Then you describe the budget, yeah, how much money you need to implement this project, and you can go to the details of the budget. So then describe the risk. It's important to talk about the risk and how you are going to mitigate those risks. So describing the risk can be tricky, but go ahead, describe them. And the most important thing is for every risk, then describe the way you mitigate. Next. So in your proposal, it's important to know that whatever you write, it's a commitment for the future. You would certainly you would like to build the trust with your donor. So if you are committed to do something, you should do it in the time you committed to do that. So when we come to the proposal, so when your proposal is approved and contract assigned and you go to the implementation phase, so you know that you committed, for example, to submit periodic reports. They could be monthly or 3-month reports, etc. You will have some monitoring to do, and make sure that you are online with your funder's template. If the funder has template to submit reports, it could be like financial report, narrative report. So just be careful and respect to the funder's criteria and the template for the reporting, the deadline for the reporting, and also like audits. You can also have periodic audits, etc. So it's important like to know that all your commitments are due and important. Next slide. Okay, I will give the floor to Erin.
Okay. Thank you, Bineta. So for your proposals, we are going through 5 tools to make your proposal more successful. So we talked about the template. Thanks, Bineta. I'm going to talk about the second tool that you all know for sure, that has many names. So an ancient name is logical framework. The new name is the theory of change. But really, on in between, you sometimes hear performance framework. In the end, we're talking always about the same thing. In fact, your project needs to be described according to a given skeleton with 5 bones, if you wish. So you start from your input, in the project. It can be finance, it can be partnership, it can be material. So all the inputs you need to conduct your activity, with, let's say, a clinical trials on leishmaniasis in Kenya again. So that's your activity. You need to describe the output of this clinical trial. So the evidence you're going to gather, the new evidence you're going to gather about a new treatment for leishmaniasis, for example. So that's really about your project. But as shown by Bineta in the template, in the usual template asked by donors, the story doesn't end there, doesn't end with the data you collect. You need to describe how the data collected will contribute to the SDG 3. So you need to describe outcomes and impact of the data that you gathered on patients affected by leishmaniasis, how their life will be better with this new treatment, so shorter time in hospital, etc., etc., etc. So don't end your story to the data collection and data analysis. You need to give, even if you're not controlling who will use your data, you need to explain to the donor the impact your work will have on the beneficiaries and here patients of leishmaniasis. So we're going to take a few minutes for very briefly for an exercise just to test you on your ability to recognize those 5 bones. So I mixed the 5 elements here, A, B, C, D, E, and to just take a few minutes to organize ABCD according to from impact to input. What is the sequence that of the pieces here that goes from impact to input? And here the example is about an organization delivering literacy courses for adults. Okay, I'll let you look at this for a minute and then we'll click on the next slide for the answer. Okay, so let's move on to the answer to this exercise. So next slide, please. And next, just one more click. Yeah, you can, yeah, exactly. Thank you. So in the logical framework, you have the input. So in the case of this organization giving literacy courses for adults, the input are facilities, the trainer, and material. So, okay. Next, the activity, it's the training course per se. Those are the activity. The output of those training courses is the fact that adults complete their literacy courses that you manage up until that point, okay? Then don't stop your story there. If you're an organization giving literacy courses, you need to describe for the funders increased literacy amongst the adults and the better jobs that adults could get thanks to your training. You see? So that's the full story that is asked with a theory of change, with logical framework, with performance agreement, performance framework. And usually around those 5 bones, you have the donors ask to specify the deliverables, the milestones, the indicators, and the assumptions behind this scheme so that they can monitor your moving from activity to outputs to impact. Okay, and on the next slide, yeah, that's the answer. Hopefully you had DCAEB as an answer. Hopefully lots of you I'm sure you did. So the next slide is an example of a logical framework from the South Korea agency. And again, it's they all have their templates, but they all ask for the same thing. So in the case of KOICA, on the top, they ask for the goal on objective, high-level objective, so the impact. And in the table, they want you to write down each outcome, each output, and linked to each output, the activities associated to that output. Okay, so if you master the little exercise we've done together before, it will be a piece of cake for you to fill this table. And as I've just said, in addition to those main bones, then the donor usually asks for indicators with a baseline on the target. So for example, indicator could be the number of patients you want to recruit in your trial. So the baseline would be 0 if you haven't started your trial, on the target might be 200, let's say. The mean of verification is the study report. The source of data is your study report. On the frequency, let's assume you can check your recruitment rate every week, okay? So this is very important to understand the skeleton of the logical framework. So next slide. As I've just said, all funders have their own way of asking for this piece of information. So here you have the example of the Gates Foundation, so much more simple. They're asking for 2 tables, one about the outcomes. You remember the outcome is really what you manage as an organization, what your team is doing. On the output is what your team is controlling. On the outcome is how the result of your work is going to impact patients' lives. Okay, next slide. We still proposing for you to use some other tools to make sure that you are producing convincing proposals. So 2 simple tools are going to follow. One you all know is the SMART. Please check that your proposal has SMART objectives. Here, I'd like you to read the 5 points on top. There are extracts from real proposals I've reviewed in your area. So, and I'd like you to identify which piece of the SMART tool are not indicated in those objectives. So are those objectives specific, measurable, achievable, realistic, timely? One dimension is missing. And I'll let you guess which one. You can use the chat as well to answer. Okay, so yeah, you obviously guessed that those objectives are quite specific. We understand quite well what each project is about. So this is really, really good. Measurable, there are measurables that indicate which drugs they're going to evaluate, etc., where, etc. Achievable and realistic, it's up to the team to judge. We cannot judge. We don't know the ins and outs of how to implement those projects, but it's for the project team to check those if what they have written is achievable and realistic. What is missing, obviously, is the timely. Okay, and it's a very important dimension of your objective. What you propose to do, is it going to take 1 year, 2 years, 3 years, 6 months? It's very important to specify that, because for the donor, they need to understand when they can expect the result of your work, on how long you will need the money for. Okay, so don't forget the timely aspects of your objectives. Next slide. A very easy tool you all know too, but which is not used as much as it could, is to, once you have written in your proposal your activities, please make sure that they are well described. Okay, and to do that, it's very simple. You read your description and check if your description covers the what needs to be done, why, who does this activity, where, when, and how. Okay, so I'll let you read this small example of activity on the left. Okay, it's very short. So here we have the what. It's an activity about a prospective study. We have little else than the what. In fact, we know it's a 3-arm study. We know it's about parasitology on clinical data, but we don't know who is going to implement this project. We don't know how this study is going to take place. So please describe the site preparation, how you're going to recruit patients in this trial, how the data is going to be collected, on how it's going to be analyzed, on who or how you're going to publish your data. Okay, so describe the how. It will give you credential for the activity you propose to conduct. Okay, so 5W for your activities. Next slide. The last tool, again, very simple, but very useful, is you can, before jumping into the writing of a proposal, so you identify the call, you know you're eligible for that call, and too often we jump into the writing of the proposal. This is one way of fundraising, but what our advice with Bineta today is just take half a day to plan your work. Check the guidelines as explained by Bineta. Check the guidelines the donor has provided to make sure you gather the right information on which information is missing and you need to produce. Check, get the background information about your organization, like mission, vision, your description of your project team, gather all the, I'm sure you have really 50% of the information is already written somewhere in your organization. So get that information, don't start from scratch. It will save a lot of time. And in terms of planning, create a retro planning, meaning you have your end date when you need to submit and walk down the timeline to allow time for revision. I would say at least 2 weeks. Aim for the final draft to be ready 2 weeks before the deadline. So you have time to circulate your draft and improve it. Okay, time to write, of course, the proposal and on time to plan. So retro planning is very, very important to allow time for the revision. Sometimes you can have external stakeholders revising that you trust, who can provide advice to read your proposal and provide some useful advice. I would say the more it's revised, the better your proposal will be. Okay. And before you send it, with Bineta, we propose you to check 6 points. First, is the language used in your document in line with the vocabulary, the words used by the donors? Is the content precise and relevant? Sometimes you tend to provide too much details about some scientific points of interest to you. when you revise your work, ask yourself, is this one page about scientific detail very necessary for the donor? Remove the piece of the text that is not relevant to the donor's questions, okay? Third, is the logic of the ideas presented in my document easy to follow? Don't forget, as stressed by Bineta, it's a very competitive exercise. So the way you present your argument needs to be very easy to follow. Check grammar on spelling mistakes. That's part of sending a professional document. Check the style and the layout. It takes a bit of time, especially some donors that restrict the number of words you can give by sections. So the layout and the presentation can take easily a few days just to make sure that the end product, once you extract your proposal from their submitting online system, is readable, on full, on nice, on convincing. Okay. So use the plan, write, revise process to really enhance your work. Next slide, and back to Bineta.
I'm sorry. Okay, thank you, Erin, for everything. I think Erin almost talked about everything, and we are almost at the end of this presentation. I was saying that I saw a message reminding us the time is running. So I will finish this, we'll finish this presentation with the elevator pitch, which is very important specifically for the type of project people can have in this live webinar scientific project, which usually are very big project, a lot of scientific information, and it can be challenging to be able to summarize and to talk about your project to someone, to a potential donor in 5 minutes. So it's a big challenge. That's why we are inviting you maybe to try to practice elevator pitch, which is a summary of your project in 5 minutes. So it's very challenging, I know, but it's very helpful. You will feel at ease, and it will help you a lot. If you go to a live webinar, you can talk quickly about your project. If you go to an event, you meet a donor or you meet someone, you can talk about your project in 5 minutes and cover pretty everything. So the elevator pitch will include one sentence in your organizations. What is the name of your organizations? 1 or 2 sentences about your team, 1 sentence about your project you are presenting, and 1 sentence about the objective of the project, 1 sentence about the expected result and impact on the beneficiaries, 1 sentence about the budget. So this is pretty a way to quickly summarize the key elements of your project and to be able to deliver this speech to a potential donor that you meet in the elevator, or you meet taking a coffee, or you meet during an event. Yeah, time is precious. People are very busy. So for you, it's important to be able to deliver a very relevant summary of your project that you have in mind and that covers the key aspects of your project. This is the objective of the elevator pitch. And as described, I would like to suggest to you, if you don't have an elevator pitch, to start working on 1 elevator pitch, 1 very short summary of your project to tell anybody you would like to inform about your project. Next. Okay, so this slide summarizes everything that Karen and I talked about. Just it's a reminder. It's a reminder of like the 5 steps you go through to start fundraising for your organizations, starting with the definition of your project, going to like the identification of the donors and finding like some prospective donors for your organization. And for that, remember like the tools that Karen talked about, where you can go to find like potential donors. She talked about the Sdg Financing Lab, etc. So remember that. Then the third step is to check calls for proposals, the funders' websites, as Karen showed in her presentation. And then you plan time. She just talked about it. Take some time to plan very well the writing of your proposal, and then you're ready to go. You're ready to go. So I know that time is running, and this will probably bring this presentation to an end. Maybe just to say a few words on the graphic on the right hand, identify, secure, enhance a relationship. What we can put this is in identify source of funding and to have a target list. In the section secure, you will put together information on your donor to know who your donor are, etc. And to communicate with your donor. Enhance a relationship is all about communication, keeping like contact and maintaining contact with your donor. And this is important because it will build trust between you and your donor. Next slide. So this brings this presentation to an end. Thank you very much for your attention. Thank you for being here. I will give the floor to Karen if she wants to say like a few last words.
Thank you, Bineta. Just to say that Bineta and I, in 3 weeks, will give a 5-day training about this topic in Dakar. But we are really pleased today to share the basic, the fundamentals of fundraising with you. So thank you for your time. We are open for questions if Professor Mekonnen and Dr. Mukhiv want, as you wish.
Thank you very much, Ms. Karen and Bineta. It was very engaging and very useful fundraising tools. You have really made a very good conclusion, and the illustration, the way you hand over the training between the two of you was not boring to me, and I hope to most of the participants as well. So I think the assignment we are going to take is to exercise elevator pitch at the end of this conference, or at the by the end of the day. It was very fascinating. The 4 aspects of fundraising tools, the types of funding, to look at the right institutions, companies, and grants, very useful, and how to identify, and the search for the call for proposals, and then to increase the chance of being funded. So these are, you have really elaborated the 4 very important subtopics or parts of the fundraising tools, and I benefited a lot. Sometimes we take things for granted, but when you hear it from the experts and those who own funds like you ladies, really it helps upcoming researchers how to look for the right source of funding, and also to look for the right calls for proposal and increase the chance of being funded. Thank you very much indeed. I don't see anything on the question and answer or on the chat, but I can open the floor for a couple of maybe comments or questions or feedbacks from the audience, from the participants. Thank you very much. So, virtual meetings are tricky. How long do I wait? A couple of minutes? Okay. I don't see any. Yeah? Is there anybody?
Yes.
Yes, please. Who is
This is Simon Bolo from Nairobi. Hi, Yalem. Hi, Karin.
Simon. Hi.
Thank you very much for such a wonderful presentation and training. I think it is a very, very useful training, even though it was quite short. We are hoping that at some point, maybe we can get a more focused training, especially for our LEAP membership, now that we are thinking of transforming into a bigger platform. So we'll be getting in touch with you so that you can have a more focused training. Thank you very much.
Thank you very much. So see you soon, physically. Okay. Now we move to the next presentation. According to the program, I have my friend and colleague, Professor Joseph Ollovo. Are you ready to present? Let me introduce you very briefly. Joseph Ollovo is a professor in the Department of Immunology and Molecular Biology, College of Health Sciences, Makerere University of Kampala, Uganda. Is currently co-chair of LEAP. He's a veterinarian with a PhD in immunobiology. His studies spanned over the years on the immunology, immunopathology of diseases of poverty, particularly leishmaniasis, trypanosomiasis, and tuberculosis in the mouse, monkey, and man, with emphasis on drug and vaccine development. In partnership with the LEAP members, he continues with studies on clinical trials for anti-leishmaniasis drug development. Now, very brief introduction. I call upon Joseph Ollovo to present his presentation, his talk. Thank you very much. The floor is yours.
Thank you, Yalem, for that very good presentation, introduction. I hope I'm waking up faster. And good afternoon, everyone. It's a pleasure to all of you again. And my presentation this afternoon is fairly light. It's a story which varies from country to country, but I think it's worth sharing. The topic of the discussion is effects of COVID-19 on visceral leishmaniasis activities in Uganda. This was our experience, and as I said earlier, it's a story which you need to hear from our angle. This presentation should have been made by my colleague, Dr. Patrick Sagaki, who is a site investigator on the DNDi clinical trial. But as I was coming here personally, he requested me to present it on his behalf. I accepted because Ahmad Hospital and Makerere University shared this presentation, making it up. So can I get the next slide, please? COVID-19 or SARS-CoV-2 was first reported in Wuhan, China in December 2019. It was immediately declared a pandemic by WHO in March 2020. In Uganda, the first case of COVID-19 was reported in the same month. In an effort to stop the disease from spreading in the country, a total lockdown was instituted by the government for 60 days from April to May 2020. There was another second lockdown for 42 days recently, from June to July, to try to keep out the disease. However, the disease keeps on with high morbidity and mortality. There are high financial losses. There's anxiety, panic, uncertainty, psychosocial issues, job losses, closure of schools. Today in my country, schools have not opened for 2 years. Quite a bit of teenage pregnancies and marriages and all sorts of ills associated with it. My association with the Scientific Advisory Committee to the Minister of Health on COVID has helped me a lot to try to understand more about the disease. But of course, being a common infection, a lot of us have naturally had interest in it. Now, with all the ills above, COVID-19 had some impact on VL activities in Uganda. Also, there's also impact on NTDs and also other diseases and health services. But the focus of this talk is on VL. We still have very little knowledge about the disease. Though a lot is being done worldwide, we still don't know much about it. The good hope is that there's a vaccine which is available for emergency. Now, COVID-19 has unlocked the world potential in all spheres. And this is the first time in the history of vaccines, which has been developed within a very short time, using some of the most advanced technology ever thought of. And part of this technology has been locked up for 10 to 15 years. Can I have the next slide? Now, this is the country I'm talking about, Uganda. Let's focus on the northeastern side of the country with the pink color. Those are the districts. There are about 9 of them, which are endemic for Leishmania donovani. And to the eastern border is the Kenya. To the northeast of Uganda borders the Turkana, which is also endemic for VL. And to the southeast borders the Pokot region, which also extends into the Pokot County, which also covers BL. To the north is South Sudan, and we don't know very little, but of course, VL in Sudan is on another level altogether. Can we move on? Now, this is Samudat Hospital, which is located about 400 kilometers from the capital. And it's located in the disease endemic area south of Karamoja region, which has about a population of about 2 million. It's a 120-bed PNFP hospital and was built in 1952 by the missionaries, although the government these days offers some financial support. This is the only hospital so far in Uganda which offers curative and preventive health services and is the only treatment center. But the story might change soon with the advent of new funders. And the hospital runs heavily on DNDi support. Our aim was to determine the impact of COVID-19 on the VL management, research and training programs. This was to enable us plan for VL activities during COVID-19. Much as we didn't know, and especially even today, we don't know for how long COVID-19 is going to persist for. We collected our data from the Amudad hospital records and also from discussion with hospital administration and staff. And we used data from 2018 to 2021. Can you move on? Next slide, please. Now, our results show that there are still up to now, we don't have any case of VL COVID co-infection. reported in the country and also in Amudat. However, the number of patients, staff in the hospital tested positive for COVID-19. The overall effects on the patients is that outpatient numbers reduced during COVID-19 compared to pre-COVID. This is 2018 numbers compared to 2020. Interestingly, there was an increase in VL cases up to about 133 in 2020, which is a COVID time. Versus, so an increase of about 40 cases during the COVID. This is rather conflicting because we would have expected the cases to go down. Now, as you can see from this bar graph, the orange color shows the VL cases during COVID, and the blue is BL patients in the non-COVID era. Next. This again are the monthly variations in the number of patients of VL from January to December. In 2018 and 2020, the orange color is the VL patients in COVID and the blue is in non-COVID time. Next. The effects on the clinical trial and research. COVID came at a time when a clinical trial, which has just ended on methotrexate PM combination, was at the tail end, and it was difficult to trace the participants who had come across the border because there was a border closure, and monitoring had to be done on Zoom links, and Amudat being in a remote area, it was difficult at times to get some clarity online. So they had to move to a town where the connections were much stronger. Some activities, funded projects and others were also delayed. Next slide. Next slide, please. On the training, we normally get students from the London School of Hygiene and Tropical Medicine. These are diploma students who come for a short stint at Amudat. They could not come for 2 years. They just started wondering whether they could come next year. But we are not sure about the COVID situation. Our local students could not travel to the field because of restricted inter-district travels, curfews, and all institutions of learning had been closed. They just contemplating opening them. Now, why are the apparent increase in BL cases during COVID time? Active case funding in Amudat is routine. We've always had it. Overall, outpatient numbers decreased during COVID-19 because public transport was locked up. There was curfew, there was lockdown. Now the number of VL cases increased due to active case funding from communities despite the COVID-19. This is because the ambulance was allowed to move as usual because we needed to give services to the community even at difficult time. And we're very grateful to DNDi for having supplied Amudat with PPE this time to protect our staff. They send their best, and there were many thanks. And the apparent increase in the number of patients was fairly high, but this is observatory. It was 40 patients more compared to the previous years. Can we move on, please? Finally, COVID is still with us, and we're not sure for how long it's going to go for. But the literature tends to point out that it might turn out to be a seasonal infection, just like flu. This is about the latest literature from Spain. And we need to give more observations with larger numbers of people A better structured questionnaire should have helped a lot in trying to get more information from the community. One hope is that there's vaccine availability, which could change the picture of COVID-19. I would like to thank DNDi, IDCP, and Mukhiv University, Kampala. These are some of the patients from that hospital. You can see young people. Thank you very much for your attention.
Thank you very much for this brief and informative presentation. I see. Okay, can I read the question? Thank you, Prof. Olobo. Has COVID had an impact on VL supplies? This is the question from Marta Gonzalez-Danz.
Can I answer straight away? Or
yes, please. Yes, please.
Yeah. Thank you for that question, Marta. I'm happy you're online. We shall talk more about the students coming over. It has not had much impact on supplies, and that's the good part of it. Because some of that supplies, they were stopped quite a while, and we've been using that.
Okay, thank you very much, Professor Joseph Ollobo. Okay, I don't see any question written on the Q&A. If there is any other, maybe one question, if anybody's interested to have an intervention. A question from where?
A short,
okay.
Yeah.
Yes, please.
A short,
yes, please.
A short question to Professor Olobo.
Yes, please.
Have you actually seen co-infected patients, and how, if so, how was it? I mean, was the management really complicated, or?
I've not understood the question.
Did you find co-infected patients with VL having COVID, and what was really the management?
Good luck.
Good luck.
We didn't find any cases of VL COVID-19 co-infection. But if there were mild asymptomatic cases, those could have gone undetected. So we basically think there was no overt disease of COVID-19 in VL patients.
I ask this because we found at the very stage of the pandemic, we found a patient with COVID, also having leishmania, and in a young man of 18 years old. And it was devastating. I mean, the patient died within 3 days of diagnosis. And it was quite complicated. I can't describe the details here, but that has happened.
So
okay,
I think
thank you very much. Do you have anything to say about it?
Yeah, I was just summarizing that we need to keep on a lookout because COVID is still with us. And as time goes by, there could be changes in the numbers and numbers of co-infections. So it's something to look out for.
Okay, thank you very much. Now I leave the floor to my co-chair, Dr. Erin. The floor is yours. Thank you very much.
Thank you, Professor Allen, for the excellent chairing of the first part of the session. So we'll move on to the second half of the session as we move towards the close of the conference. But before I introduce our next speaker, just a reminder to the participants to continue tweeting using the hashtags leishmaniasis, Africadia, and B10CD. So please continue tweeting. Also, just to remind you to keep the Q&A box active. So please feel free to put in your questions on the Q&A. We'll have a moment to respond to any questions after next speaker. So just as a follow-up to what Professor Olobo has presented, so we'll keep it on COVID. I have the pleasure to introduce our colleague from Africa CDC, who will be speaking to us about COVID-19 in Africa, where we are, lessons learned, and what more needs to be done. I have the pleasure to introduce Dr. Abdulaziz Mohammed. Dr. Abdulaziz Mohammed is the head of Division Disease Control and Prevention and acting head of the Division of Surveillance and Disease Intelligence at the Africa Centers for Disease Control and Prevention at the Africa Union Commission, Addis Ababa in Ethiopia. Dr. Mohammed is also the co-chair for the Infection Prevention and Control Technical Working Group for Africa CDC, Continental Response to COVID. He's an MD with an MPH and is a fellow of the West Africa College of Physicians. He's also a fellow of the Africa CDC, Africa Union Fellowship for Leadership in Public Health in Africa. And he's also a fellow of the Chatham House Africa Leadership Program. Before his current appointment, he was previously the principal medical epidemiologist in Africa CDC, where he led the first regional initiative to strengthen public health in Africa. And Dr. Mohammed is widely published. Dr. Mohammed, welcome very much. We take this opportunity to give you the floor to give your presentation. I believe you will be projecting your own slide, Dr. Mohammed.
Thank you, Chair. Thank you for the wonderful introduction and the kind words. Let me see how yes, let me share the screen.
If you can put it on slideshow, we can see your presentation.
Yes. Is it on? It's on slideshow now.
Yes, thank you.
Okay, thank you so much. First, let me thank the organizers of this excellent conference for inviting Africa CDC, and Dr. Ahmed, the Deputy Director, asked me to come and make this presentation precisely because we have in the new Division of Disease Control and Prevention a home for neglected tropical diseases and other endemic infectious diseases on the continent. So, but I'm here to discuss lessons from COVID-19 pandemic in Africa and then Africa CDC response. Chair, I have 20 minutes, I was told.
Yes, you do.
Okay, thank you. So we'll start with the lessons we've learned so far. What are the Africa CDC response to this pandemic? And then the key takeaways that we need to learn as a continent in the response to pandemics and outbreaks and subsequent public health events that can happen on the continent. I'll take you through 8 different lessons that we've learned. So the critical thing here is that the role of political leadership and cooperation within the African Union. We need to have very strong political leadership and within the African Union, if we are ever to be able to respond effectively to any pandemic in the future. This we've learned. And then we need to use all the resources we have on the continent, harnessing all the African Union assets, including that of NEPAD, economic affairs and transportation, in all our response to the COVID-19 and other pandemics in the future. So lesson 1 when it comes to pandemic response on the continent is coordination and collaboration is key for the whole of the continent. Africa CDC, even before the onset of the outbreak in Africa, when it was still outside Africa, in China, in Europe, we convened a meeting on February 22nd, 2020 in Addis Ababa and in Ethiopia. The essence of that ministerial meeting is to bring all the Minister of Health and for us to develop a joint African Union, Africa CDC strategy then just in case this virus comes to our continent. We knew in Africa CDC that the virus was coming, and we needed it to have excellent procedures and processes to be able to manage this. So that's the very first lesson we learned on the continent, that we really need to have a coordinated Africa response to this pandemic. So what did we do? We set up the Africa Task Force for Coronavirus, which all the areas that we knew would be key for the continent to have a coordinated response, areas like surveillance, diagnostic, infection prevention and control, clinical management, risk communication and community engagement, supply chain and stockpile research. We set up technical working group in all of these areas to coordinate, to cooperate among the various stakeholders, to collaborate between WHO, Africa CDC, UNICEF, and all other agencies that will be essential for the continent to be able to respond adequately to this pandemic, and most importantly, to communicate, because we know that we need to be able to communicate across the continent for this continent to stand a chance of preparing for and be able to take care of this pandemic. Lesson 2 that we learned: Africa CDC and the continent needs a network of very strong national public health institutions. Meaning, in all the countries of the continent, we need people who can, institutions who will be strong and be able to respond to any public health event for the continent. So if you look at Nigeria, for instance, we have the Nigeria CDC. In Ethiopia, we have the Ethiopian Public Health Institution. In Liberia, we have the Liberian National Public Health Institution. But not all African countries have this strong institution that can be able to coordinate, to cooperate, to collaborate, and bring strong leadership to pandemics, outbreaks, and other public health events. So one critical lesson we've learned is that we need to support and strengthen national public health institutions across the continent. Lesson 3, we need to develop continent-specific guidelines. Africa has 5 regions, but we are different from other regions of the world, meaning we need to contextualize different guidelines in different parts of the world to Africa. We need to be able to look at it from the point of view of Africa. Let me give you an example. We have guidelines from other parts of the world which says that you do not use air conditioning or something, but when you look at it in the context of Africa, humidity and the heat will not allow a health worker to put on the gown and work for more than 5 minutes in excruciating heat and excruciating humidity. Therefore, we need to look at issues and be able to contextualize it based on the current realities on the continent. We differ from other parts of the world, as you will see in the subsequent presentation. So lesson 4 that we've learned is that the continent political leadership needs to guide the pandemic response. We need our head of state, and this is exactly what they did. We need our ministers, and this is exactly what they did. On the 8th of May, 2020, African Ministers of Health endorsed a strategy to fight COVID-19, underpinned by the processes of the prevention first, if you can, monitor, and then treat. And we call that strategy the Joint Continental Strategy. And we were able to use this strategy, as you will see, subsequently in the fight against this pandemic on the continent. Now, it became right at the onset very clear to us that Africa needs local diagnostic manufacturing. And it is a health security for us. Because as you will see, Africa CDC was able to build quickly the capacity of the continent to diagnose COVID-19. However, it was a great threat when we could not countries could not get even the basic diagnostic tests to do COVID-19. They have the capacity, but they cannot do the test because they don't have the diagnostics. Africa CDC had to run and really be able to begin to set up modalities and mechanisms to ship these diagnostic tests from another continent to the continent of Africa. Can we do this the next time? Then we are not sure. Therefore, we have to begin to look at how to produce diagnostics locally on the continent. If you look at from February, when the first case was announced in Egypt, up to August, the test rate in Africa was so low because of this lack of this, until we were able to really rally using the partnership for accelerated COVID testing, starting from around June. That is when you can see the test we were able to increase up to where we are now, over 70 million tests are conducted on the continent. So in January, when we had the first cases in Asia and in Europe, Africa had absolutely no capacity to make a diagnosis of COVID-19. In February, 2 countries, South Africa and Senegal, developed that capacity. Africa CDC moved in, supporting different countries on the continent, virtually the whole continent, with over 1,170 lab personnel training, first in Senegal, that have the capacity, then the second training in South Africa, and then we did more. These are face-to-face training. After it hits the continent and we were able we could not travel because of the lockdown, we turned to doing online training. And luckily, because we've already trained people in country, for most of the country, we're able to use those people to rapidly build the diagnostic capacity of African countries, so much so that by August, virtually every country in Africa could make a diagnosis of COVID-19. I'm talking about August 2020. And this was really key in being able to control and monitor this pandemic. We've distributed over 40 million kits, and out of the 60 million tests that were done on the continent. Importantly, again, we need to be able to produce vaccine locally on the continent. If you look at right now, 99% of all doses we are using on the continent are imported. All 99%. And if you look at the map on your right, Africa has one of the least vaccine penetration in the world. You could see that most of Africa is between 0 to 5% or 0 to 5 million vaccinated is white compared to the rest of the world. COVID-19 has exposed Africa's dependence on vaccines being brought from abroad. This is not a way to have global Africa health security. If we cannot produce vaccines, we will always be last on the queue, meaning we need to begin to produce vaccine locally on the continent. So the Africa Joint Continental Vaccine Development and Access Strategy in August 2020 was developed, and we had very strong support from our head of state. The head of state and government renewed their call for member states to contribute towards the AU COVID-19 Solidarity Fund, which is critical. Hello, can you hear me? Hello?
Yes.
Okay.
Yes.
Okay. Okay. Which is critical for the effective implementation of the continental strategy. They endorsed the COVID-19 vaccine development access strategy as presented by the director of Africa CDC. In a conference, we had over 40,000 participants from 70 countries, and more than 13 head of states were in that conference, where we looked at the need for expanding Africa vaccines manufacturing and thereby having global Africa's security when it comes to pandemic response. And the Africa CDC launched the partnership for the Africa vaccine manufacturing, that's PAVM, to ensure Africa has timely access to vaccine to protect public health security by establishing a sustainable vaccine development and manufacturing ecosystem on the continent. We had very strong support from our head of state, especially President Kagame. The 7th lessons we've learned is that local manufacturing of PPEs, therapeutics, and medical equipment is a health security challenge. We have talked about local manufacturing of diagnostics. We've talked about local manufacturing of vaccines. We are now talking about local manufacturing of infection prevention and control materials. These are critical. If not for the humanitarian corridors Africa CDC created at the heat of the lockdown, and Africa did lockdown, we will have, in fact, at a point in time, countries were running short of simple face masks was not available, and even elective procedures could not be done because face masks was not available. This has to stop because we need to manufacture. If we have countries in Africa manufacturing these things, we will not run into the kind of serious trouble we ran into some time ago. Based on that, Africa CDC had no choice. We had to distribute ventilators, oxygen concentrators, thermoscanners, infrared thermometers, face shields, face masks, gloves, and many other things just to ensure that our continent was able to respond adequately even during the lockdown to the pandemic and protect our citizens. So lesson 8 that we learned is the urgent need for the Africa public health workforce development. As you see subsequently, the workforce development on the continent is seriously challenged, and we need to ensure that we improve on that. The Africa Epidemic Service is what we are proposing, where we have the FETPs, the laboratory leadership, and public health informatics. Now, as you can see, for field epidemiologists, on the continent, we have nearly 2,000. We need 4,000. For frontline epidemiologists, we have just 5,000. We need 25,000. So a gap of 20,000. So these are some of the areas where we felt we really need to begin to respond to the workforce need of the continent. So far, as of August 2021, we've trained 211 surveillance specialists, 930 IPC, 30 case management specialists, and 369 risk communicators. And we continue to support member states to ensure that we build this component. We are doing the African Union Commission Kofi Annan Global Health Leadership Program, where we want to train the next generation of leaders when it comes to health on the continent. And it's a fellowship in public health leadership program and a virtual leadership academy because we found ourselves during in the pandemic period. So what are the things Africa CDC had to do to respond to this COVID-19? We've had to do a series of different initiatives and programs tailored towards the natural history of this pandemic. At the beginning, we did the partnership for accelerated COVID testing because that was what was needed. We needed to know if the virus hits the continent. We needed to be able to do testing so that we can do contact tracing and other things. So we were able to put in a process that quickly accelerated testing on the continent. We needed a response fund because we needed to supply countries with what is required. And we needed a situation in which we can ask our member states to go and buy this thing using pool purchasing. Because if they had to go individually to meet the manufacturers, there would be problem of middlemen. So we set up the African Medicine Supply Platform where a member state could go and get IPC materials at manufacturers' rate so that they can be able to respond effectively. And of course, we set up, like I said, the African Task Force on Coronavirus that coordinates all our response. We realized quite early that vaccination will be the key to ultimate control of this pandemic. So we set up a Africa CDC vaccine strategy for COVID-19 vaccine, which include the Africa Vaccine Acquisition Task Team, the African Vaccine Delivery Alliance, and of course the Partnership for Vaccine Manufacturing on the continent. So Africa CDC still, so not Africa CDC, the African continent is still facing a third wave. And to combat this third wave, we've deployed teams to Seychelles, Namibia, Cameroon, and Sierra Leone. We've supported with over 700,000 USD worth of equipment and supply to each of these countries. And currently we are recruiting for national rapid response and community health workers to send to many countries on the continent. And we need to know the variant that is circulating on the continent. And therefore we are supporting member states with reagent and equipment necessary to do pathogen genomics. Africa CDC continue to support the third wave. This is just to show you our teams in Zambia, teams in Namibia, teams in Cameroon, and of course the teams working in Sierra Leone supporting this. So far, Africa CDC have deployed over 18,000 community health workers to over 28 countries on the continent, and we've deployed over 200 rapid response volunteers, that's senior people like epidemiologists, to over 25 countries on the continent. We've talked about the Africa Medical Supply Platform that we set up so that countries can get pool purchasing of critical supplies necessary for pandemic response. We've worked in setting up trusted, we know right from time when even countries were locking down that we need to reopen. And if we reopen, we have to reopen in a safe way. So we set up the Africa CDC Trusted Travel program, which set up a trusted travel portal where countries can feel safe in reopening their economy, reopening their air transport, so that commerce and trade can resume. So that platform has the cross-border travel solution, vaccine record management, and then vaccine allocation and administration schedule. We're still rolling out in different countries. I think in the last count, I know that we have rolled out in about 7 or 8 countries in Africa. We need vaccines for vaccination so that we can save lives. That's for sure. The aim of Africa CDC is to vaccinate 60% of Africa. That is the minimum we require to prevent Africa being a place where we will begin to have variants popping up left, right, and center. Meaning we have to vaccinate a minimum of 800 million Africans, and with giving a total of 1.6 billion vaccines. So different kinds of vaccines are coming to the continent. We have those coming through the COVAX facility. We have the one Africa CDC and the African Union is using through the AVAT facility, and we have some coming through the bilaterals and multilaterals. However, we know that our target is to reach 60% coverage. The AVAT doses, which we are championing, is supported by Afrexim Bank facility, and additional partner support is also coming in. So where are we today? Compared to the rest of the world, we are still very, very far behind. China has gotten to 76%, 71% for fully vaccinated. United States is at 54% fully vaccinated. Europe is at 51% fully vaccinated. India is at 15%. The world average is 32%. Africa is still at 4%. So we understand that in Africa CDC. I'll tell you subsequently what we're doing next to ensure that we're able to change the narrative in that area. There are some countries that have really been able to make success. Morocco is at 50% now, Mauritius at 61%, and Seychelles at over 71%. So we are vaccinating in Africa. You can see what's happening in Zimbabwe, you can see it in Rwanda, but we need to do more. So the African Union announced the rollout of 400 million vaccine doses to the African Union member states and the Caribbean, supported by what we call the AVAT projects. So the AVAT project is now, or program, let me say, is now sending vaccines are arriving in masses in different countries in Africa, supporting the ones that are already on ground. So here in Togo is receiving, Botswana is receiving, Cameroon received, Ethiopia is also receiving, Lesotho has received, Nigeria receiving, Zambia, and ladies and gentlemen, why are we so sure vaccine is what we need? If you look at this data, 99.2% of COVID-19 deaths in June were in unvaccinated population, meaning we are losing people as long as we keep our vaccination at that low level of abysmal 4.2. So something needs to be done. First, what needs to be done is we need to build the capacity of African countries to manufacture vaccine. So Senegal is doing something, Morocco, South Africa, and Egypt, and Algeria are all doing something. We hope to be able to scale up all of what this is happening so that whenever the next pandemic, and we know there will be a next pandemic, Africa will be better prepared when it comes to local manufacturing of vaccines. Then Africa CDC is partnering with MasterCard in an unprecedented 1.3 billion USD investment to be able to get to that goal of vaccinating 60% of Africans by the year ending 2022. So it's a massive undertaking with massive funding. We want to be able to reach that herd immunity by the year 2022. And we're doing it in partnership with the MasterCard Foundation. So ending COVID-19 pandemic will require a global collaboration and effort ensuring that everyone has access to vaccine, tests, and treatment. If no one is safe, no one is safe until everybody is safe. We all know that one. We've been saying it. And Africa CDC is really committed to that. So the Saving Lives and Saving Livelihood program benefits access to COVID-19 vaccination to millions of Africans, strengthen member states' vaccine rollout capacity. Will be supporting member states to support in rolling out COVID-19 vaccine capacity. We want to strengthen African institutions to drive broader impact and ensure national, regional, and global health security. So, in concluding, distinguished participants of this conference, we say that no complacency, no prevention fatigue. We need to intensify public health and social measures against COVID-19 by continuous political leadership to implementing the following: support COVID-19 vaccine access, delivery, and vaccination campaigns. Enhance community engagement for improved usage of face masks and hand washing measures. Avoid large gathering and crowded events, and support community surveillance. Africa CDC in the coming weeks will be announcing a huge undertaking to support member states in an enhanced community surveillance process that we hope will be able to help move us from where we are today to a more enhanced monitoring of where we are with the pandemic and how we need to respond. Then, of course, ensure adequate supply of PPEs. And without vaccine and vaccination, the COVID-19 situation will get worse before it gets better. Any part of the world that think it's safe is safe. If another part of the world is not safe, we cannot say we have collective safety, because this virus, as we know, is an RNA virus and can easily mutate to another form. And that is why we need to address this as a global health security where everybody can be protected. We are slowly getting into a state of endemicity. This can be averted if we act decisively. New COVID-19 waves is ongoing. We cannot rule them out. The presence of variants and low level of vaccine coverage means social harm and devastating economies. If you remember what happened at the height of the lockdown, many African countries went into recession and lost billions of dollars, which means that health security is actually economic security. Financing of vaccine and vaccination is root to overall economic recovery. We need to make investment in that area as a continent. So we need to strengthen continental and national public health institutions. We've seen it. We need to strengthen public health workforce, expand local manufacturing of vaccines, diagnostic and therapeutics, and have respectful partnership. Then we need to build back better, bolder, and bigger. Thank you, ladies and gentlemen. My apologies for maybe taking a bit longer than necessary. Let me hand over back to the chair.
Excellent. So thank you very much, Dr. Mohammed. So I think first, before I say anything else, I hope that you will stay with us to the end of the closing session. We have a photo session and we wouldn't want you to miss it. And so for the rest of the participants. So I think it's impossible to summarize your presentation. And we'll have a few minutes for anybody who has any question to respond to you. But I think in terms of the takeaways, you said some critical things. One, especially the power of leadership, the forces you mentioned, the coordinate, cooperate, collaboration, and communication. And then you also talked about the critical need for Africa continent to develop the capacity for development, not just for vaccinations, but also therapeutics and also diagnostics. And it's impressive to see the massive amount of effort and coordination that Africa CDC has led with the participation of African governments. Totally impressive. We hope that as we move forward, we can see the same sort of political capital and collaboration towards addressing the issue of NTDs, which, as you know, part of the focus of this discussion is the neglect around not only leishmaniasis, but other tropical neglected diseases. So I want to take this opportunity to see if there is any question, if anybody might want to pose a question to Dr. Mohammed, either within the room or online. But Dr. Mohammed, I have a question for you. And this is perhaps more towards the long-term thinking. How do you see some of these platforms that the Africa CDC has developed evolving over time, hoping maybe we'll see a stabilization of the COVID situation, but how do you see some of these initiatives becoming more institutionalized and evolving to address some of the other issues that, that Africa is facing? Over to you, Dr.
Excellent chair, Dr. Irene, and I will address it together with your first comment, which is what Africa CDC is planning to do for neglected tropical diseases. Let me say ab initio that when the African head of state first thought of setting up a continental public health institutions, they were looking at addressing issues that were affecting Africans on a day-to-day basis. So we're talking about endemic and neglected tropical diseases. But they were looking at, obviously, the big ones, the HIV, TB, and malaria, but also the neglected tropical diseases like hepatitis, like leishmaniasis. These are diseases that have been with Africans for donkey years and have not been given their due attention. So that was in Abuja in 2013. However, the events in 2014, the Ebola event, shifted the focus a little to global health security agenda based on the lessons we've learned from the 2014 Ebola outbreak in West Africa. And so when Africa CDC came on board, the focus was to have a strong surveillance, event-based surveillance, laboratory network, a strong emergency preparedness and response division, and strengthening the national public health institution. Now that we feel we're doing that and we're doing very well, right in the middle of the pandemic in 2020, Africa CDC set up the Division of Disease Control and Prevention. Under that division, we have a unit dedicated to endemic and neglected tropical diseases. And we are in the process of building the capacity of that unit. We have about 4 people already working in that unit with several strong candidates coming very soon to join that unit. So we are looking at how we can change the narrative when it comes to neglected tropical diseases and endemic infectious diseases. Let me assure you on that. Again, some of this platform that we've produced to respond to this pandemic, one thing we've learned is that we must institutionalize some of the lessons we've done. Look at the African Medicine Supply Platform. We're moving from using it only for the COVID-19 to use it, we've used it for vaccines. We hope to use it, the pool procurement for other aspects where countries can come and get some other medication to treat other endemic and infectious non-neglected tropical diseases. We're looking at how to ensure that we are able to, for instance, the community health workers that I told you that we've deployed, we are now moving it into a unit in the Division of Disease Control and Prevention that will ensure sustainability of all of those interventions, not just for the COVID response, but also for other response, including supporting universal health coverage and other things like prevention and control of some entities like hepatitis, which we're already working on, and then of course the big ones, HIV, TB, and malaria. Thank you. Over to you.
Thank you, Dr. Mohammed, for that. So we have one additional question on the chat. I'll read it to you, and then we'll go to you, Professor Olobo. So one question from one of the participants is, what is Africa CDC doing in relation to therapeutics for COVID-19? I think you highlighted it, but if you could respond to the question.
Absolutely. We are supporting local manufacturing of diagnostics, therapeutics, and vaccines and other infection and prevention and control strategy. To tell you, if you look at the massive investment we're doing in the vaccine area, part of that money is set for local manufacturing of all of those critical consumables that are required for pandemic and outbreak response. Then we've supported the distribution of nearly 700 oxygen concentrators and respirators throughout the period of this pandemic. We are supporting with training. We've trained quite a number of first responders and others during this pandemic response, even when there was closed, I mean, total lockdown in most of the African countries. We continue. If you look at the last slide in that, we were able to move using military planes. Even during the lockdown, we're able to move critically needed workforce around the continent to ensure that they are able to support member states in the response. We'll continue to do that. We're supporting research and supporting African countries able to come up with diagnostic therapeutics and all sorts of innovation that will ensure that our response to this pandemic is improved. Thank you. Over to you.
Thank you, Dr. Mohammed. So I think we'll take one final question to you from Professor Ollobo.
Thank
Please
you.
Go ahead.
Thank you, Chair. Thank you, Dr. Mohammed. It's been nice listening to you. Stories around COVID-19 don't seem to end soon. I have some issues I wanted to get your opinion on. Africa, as you mentioned earlier, is very vulnerable. And we're always last in the world. Africa has very little to offer in the world. And in case of another resurgence, reemergence of another disease, if we are not lucky, we could get wiped out. I say this not with reservation, because when COVID came, why the levels of cases are going down, we can't yet explain. Now, Europe and developed countries are where they are because they put a lot of efforts, a lot of resources, and a lot of energy into basic research to lead them, to guide them. So whatever they do, they have a guidance. They have a guidance. as far as I see our sciences, we seem to be lacking the capacity building for basic biology, basic sciences, to let us, dissect the virus, dissect the parasite, dissect the plant, analyze it, and, give something for the other group of scientists to be able to do something. We have a lot of technology in young people. We've trained a lot of young people in immunology, molecular biology, biochemistry, chemistry, epidemiology, but they don't seem to be utilized, in where I would call power labs, where their energies, their intelligence, their specialization could be used. The story of Cuba comes to me very, very readily here. Cuba is now about 4 vaccines, and they've used very simple technology for COVID vaccines. They use 4, they've now got 4, and they use very simple technology, which was there 25 years ago, and which is used actually for manufacturing, hepatitis B vaccine. Now, my worry is it possible for the African Union to put its foot down, come up with a core lab, whereby, good brains can be put, whereby funding can be put. We have institutions like ILRI, whereby, for 20 years or so, they put in a lot of money and they pull the best scientists from all over the world to try to get a vaccine for East Coast fever and trypanosomiasis, but it didn't work. But I'm talking of something like that, whereby, we can put our brains together, get the best brains, and then we can be proud to offer something to the world. Otherwise, we are in a very mixed situation.
Thank you. Thanks, Professor Olobo. So over to you, Dr. Mohammed, quite a challenge.
Thank you, Prof. Really an excellent question. And let me attempt to answer it this way. Part of the problem we've had in the past in Africa is that when it comes to health, we have weak national institutions. We've had a lot of interventions on the continent, but we do not have national institutions where the memory, the learning from those interventions is warehoused. And then we can have continuous strengthening of those national institutions to be able to have a place where the kind of intervention you are talking about will come from. If you look at the US CDC, the US CDC came from what they learned about malaria very early in the 20th century we talk about now. Look at China CDC. China CDC used the response to the earlier SARS-CoV-2 virus, that's severe acute respiratory syndrome, earlier in the century, to build very strong institutions that can be able to respond to pandemic and outbreak and other public health events. In Africa, we do not, in most of our countries, we do not have this strong national public health institution. So today somebody comes for HIV, he does it, then at the end of the program, there is no institution that will keep the learning from that. We just finished eradicating or eliminating polio. Do we have institutions that can keep the learning that most of our countries have gained from doing that? So that is one, meaning we need to invest in very strong national public health institution that can have the strength and temerity to work on this basic research that you're talking about and translate it into public health action that will be useful for the future. Importantly, you touch on another area that Africa CDC is working very hard on, the area of workforce development. The kind of priorities we're pushing our people to do, are they the priorities that we need on the continent? We now know that because of this pandemic response, we do need health informaticians, we need molecular biologists, we need all of these kind of people that will be able to sit down and using basic research and form of operational research, give us the kind of evidence that institutions like NCDC, like Ethiopian Public Health Institution, needs to control this pandemic. So this is one area where Africa CDC is working very hard on. We are supporting the field epidemiology and laboratory training programs across the continent. We are setting up programs, scholar programs that will support the building of this kind of workforce, specifically tailored at the gaps that we've noticed on the continent. For instance, we are supporting people to go to Wits University in South Africa to do masters and PhDs. We'll continue to support these critical areas and plow them back into the member states so that we can in the future have these kind of things that you are mentioning. It requires a lot of investment from member states, not just the continent, Africa's CDC at the continental regional level. It requires a lot of investment, and that is why we feel the best way to do this is by advocacy to the head of states, advocacy to the ministers of health, to ensure that we build the public health institutions that can be able to respond the way you've just said in terms of building the right kind of structure, health system that will be able to contribute to what we are doing. Thank you. Over to you, Chair.
Thank you very much, Dr. Mohammed, for not only the excellent presentation, but also highlighting the additional work that Africa CDC is mobilizing everybody to do. So I think to end that session, Monique has one final comment, and then we will now switch over to the closing session after Monique's comment.
Thank you very much. Thank you very much, Irene. I was so inspired by Dr. Mohammed's presentation that I just felt I need to say something as an African woman. And I just wanted to say thank you to Africa CDC, because for a long time, the African believed they can't think, they can't do anything, and so now we are having the continent inspired. That you can think, you can work together, we can work together. Yes, we can innovate, the sky is the limit. So I think this is we really want to thank the leadership of Africa CDC and all of you, Dr. Mohammed and the team, for what you're doing for us in Africa. Just making sure that, yes, we are capable, yes, we are agile, and building institutions is the way to go, and inspiring talent in the young people. So I know we'll thank you, but I just wanted you to come from the organizing committee that really we appreciate this powerful, it was the most powerful speech I've had in a long time. So thank you.
Thank you, Monique. Okay, so we'll now switch gear just to acknowledge the very excellent presentation. So I think the last few presentations have had so many nice points. So I urge you to keep tweeting. We should be seeing tweets streaming live right now. We'll be switching over to the closing session. I'd just like to request all the participants to please hang on. We have another 15 minutes, just 15 minutes to wind up. Hang on, we have a photo session. You don't want to miss the photo session, so please stay on to the session for the next 15 minutes. So we'll now move over to the closing session. And I would like to take this opportunity. We have two speakers to close for us. And I would like to introduce our next speaker, who is not with us in the meeting, but will play a short video. Professor Ivan Velez, MD, PhD, who is the director of PESET, which is Program for Research for Tropical Diseases from the University of Antioquia in Colombia, and is also the chair of WildLish 7, who is going to spend the next few minutes to give us an update on WildLish 7. Edwin, if you can proceed. Thank you. So just to summarize the video that will be meeting in Cartagena, so I think everybody needs to start preparing for that. So, so we'll move on to the next speaker. And at this point, I'd like to introduce Dr. Jorge. I hope that is the right pronunciation. Thank you. So let me take this opportunity to introduce Jorge, who is going to give the sort of close-up remarks and also give a vote of thanks for the very exciting conference that we've had for the last 3 days. Jorge Alva is a senior advisor of the Leishmaniasis program at DNDi, a program he has directed for the last more than 5 years. He's also a full member of the Royal National Academy of the Medicine of Spain since 29th June, but previously has been a corresponding member since 1994. Jorge is an MD with a diploma in tropical medicine from Germany, and is also a visiting scientist at the University of Cambridge in the United Kingdom. Prior to joining DNDi, he worked as the medical officer in charge of leishmaniasis control program at the Department of NTD at WHO. And prior to that was a director of the National Center of Tropical Medicine at the Institute of Health, Carlos in Madrid, Spain. Jorge has considerable expertise, has received multiple awards for his research in leishmaniasis and also AIDS co-infection, and is widely published and has participated in multiple international and national research agencies and presented in numerous conferences. So I'd like to take this opportunity to welcome you to give us sort of a summary and overview of the last 3 days and also give closing remarks. And as soon as Jorge is done with his speech, I'll request all the participants to please switch on their videos for a photo moment, and then we'll close down. So please welcome.
Thank you very much, Dr. Erin. Well, a long introduction, just to say that about everything, I'm a good friend of Africa. I've been working for many years, and so then, it's always a pleasure to be with you. Actually, it's not a 3 days conference. We are embarked since a week entirely. I think it has been an exhaustive week for the team in Nairobi, preparing everything, because you know, Monday we have the meeting of the LEAP governance bodies, then the day after a workshop on access for VL drugs, and then is when the LEAP conference started. So then, first of all, I have to express my gratitude to the team in Nairobi, who has done an excellent work. But you know, we can enter in detailing thanks to everybody, and I will, because it's the moment to do this. But for me, this second conference, LEAP conference, is remarkable. I think it's a benchmark, at least for 3 aspects. Number one is the first time ever in which formally WHO and the countries are talking on elimination of VL in Africa after the experience gained in East Asia. And this is important because from now onwards, we have to think more at the regional level, you know, rather than with scattered activities. Second, is important because the substantial scientific advances in terms of treatment, in terms of immunology, and it was really, you know, a pleasure to listen directly, you know, young scientists from various African countries, from Ghana, from Morocco, of course, from East Africa. So then this aspect on how science has evolved in the African continent, I think is remarkable. The LEAP group has done an excellent job in developing new treatments for VL, also for PKDL, and also for HIV-VL. But also immunologists from other groups have been, you know, developing their own studies in vaccines. So then, control elimination. Second, how science has advanced. But also it's remarkable this conference because it's the first time in which cutaneous leishmaniasis is taken, you know, in a comprehensive manner. Has, you know, probably 1/3 of the Congress has been devoted to cutaneous leishmaniasis, something that the governments and the people and the scientists were asking for since many years. So then the Congress has been relevant, and has been relevant also because it worked really well. I mean, the hotel facilities, you know, those, I mean, to coordinate the teleconference is not an easy thing, and it was very smoothly. I was really impressed on the quality. because of course this was a challenge also for the Colombian colleagues preparing World List 7, how things can be done. And I think this was fantastic. I want to also to summarize, with some figures. The quality is the quality, and it's difficult to be put in figures, but it's also, good to know that 57 countries have participated in the conference. 22 from Africa and 35 from the world. So then this is probably an experiment for you in Africa to thinking in the World List 8. I think the power you have to attract countries and scientists is really to be taken into account. Then yes, why the Congress was good? More than 200 participants is not peanuts, considering the time in which we are. 200 participants is really, good figure. And again, the 57 countries participating. I think this is important, but this is even more important, the content, and the content has been really very, very interesting. And we have in here to express our gratitude to the scientific committee. And I cannot enumerate one by one, but the scientific committee did an excellent job. But also the panelists, the chair and co-chair of the sessions, the presenters, of course, and indeed the audience. So then we all are in the same boat, and I think the job has been excellent. The Congress started, very powerful, with the presence of Dr. Sheila Musonda from the African Union. We need the politicians now if we want, to move to an elimination strategy at the regional level. So then I really celebrate that Sheila Musonda was present, I know, representing her own boss, etc. But they are committed already. I think that this is very important. Also, with the others speakers in the opening ceremony, I think this was also brilliant to have, this bunch of personalities around leishmaniasis. And I have to say, with all of you probably, that we were impressed with Dr. Muhammad Abdulaziz from CDC. I think we have to bring him to Colombia as well. No? It's really very impressive. And so then, yes, about everything, there is always someone working hard, no sleeping at night. And this is Monique Wasuna, the chair of the Congress, supported by the scientific committee chaired by Simon Bolo. I think without them, nothing would have happened. And of course, the money is the money. And that money came from the EDCTP and also the Africa DIA project that many colleagues here have been benefited from this project, benefited and doing an excellent job, as mentioned before, and DNDi. I think we have to recognize them because to have those supportive institutions is always very, very much appreciated. It has not been mentioned that the quality was so good that we had the opportunity to discuss in the organizing committee of World List 7 to announce here three awards for the best abstracts or presentations that you consider. They have to be, first of all, young students, first authors in the studies, and you decide. But WordLess 7 in Colombia will be very happy to waive their registration fees as a bridging system between Africa and Colombia this time. So then please organize your scientific committee to make the choice of the 3 students that can be benefited from this award by WordLess 7, and make it public in the webpage of the second LEAP conference within a week or so. Is up to you, but please let know publicly who were the ones benefiting, and they will be, very especially well treated in Colombia. And that's it. That's it from my side. Of course, I cannot list everybody, in the participating, but it was a pleasure. It was a pleasure, and the good news is that in about 10 months we can meet again in Colombia. Something that I encourage you to. Because it really is a nice country, and the town of Cartagena is very impressive. Thank you very much, everybody. Thank you very much to Monique's team and the LEAP members, that at the end of the day are those that are, doing the research in the field, and to the patients. And I wish you good luck, with the hope to see you all face to face in Colombia. From my side, I can announce now that the second LEAP conference is over. Thank you very much, everybody, and stay for the photography, please.
Thank you, Jorge. I don't know whether there is a way of clapping online or wherever we are, but we should probably clap. Please put on your video. We will have I hope everybody is able to put on their video, IT. Please put on your video for a photo session. When everybody has their video on, I think the only way to show our teeth is to say leash. So are we ready? Okay, so shall we say leash so that we can show our teeth?
Does that include us?
Yes.
Oh.
Is everybody there?
Put on more lights.
Will be in the annex.
Oh,
On them.
On them.
Are you connected?
No.
Okay, so just keep smiling until the costume tells us
to stop.
It's enough.
Yes. Straight.
There are some people I don't see their faces.
These are page 2. These are page 2.
Let's say leash one more time so that the last photo can be captured.
I think we should have one crazy one where we do something crazy.
Yeah, that's a good idea.
There are some crazy people who can't.
Okay, so we are going to tend to beat NTDs. So when I say one, everybody is going to say we beat NTDs, okay?
Okay.
And then comms team, please say. So, so 1, 2, 3, we beat NTDs. Okay, once more.
They should unmute.
No?
They should unmute. So that
Yeah, we are not, yeah.
Are you unmuting everybody?
Everybody to unmute.
Okay, please go ahead. Okay, so we'll go over that again.
Other than you.
Okay, so 1, 2, 3.
We beat NTD.
Again.
Okay, so 1, 2, 3. We beat
NTD.
Okay, so thank you, everybody. We meet in Colombia.
Thank you.
Thank you.
Thank you very much. Thank you,
very much.
everybody.
Thank you very much.
Friends, good friends around.
Yeah.
Thank you.
Thank you. This was a great meeting.
Bye.
Bye.
Bye, 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 Monique. Bye, Monique.
Bye. Bye. Thank you. Bye.
Musa. Musa.
We missed you.
The Sudanese friends, bye. We miss you. Bye.
Okay. Sleep should not sleep.